What if education wasn't just about learning? What if it was about igniting a lifelong passion for excellence? Dopamine is more than a name, it's a promise to empower the next generation of medical minds. With a mission to make medical education accessible, affordable, and revolutionary, Dopamine is here to redefine learning for undergraduates. Join us in shaping the future of healthcare, one brilliant mind at a time. What if education wasn't just about learning? What if it was about igniting a lifelong passion for excellence? Dopamine is more than a name, it's a promise to empower the next generation of medical minds. With a mission to make medical education accessible, affordable, and revolutionary, Dopamine is here to redefine learning for undergraduates. Join us in shaping the future of healthcare, one brilliant mind at a time. What if education wasn't just about learning? What if it was about igniting a lifelong passion for excellence? Dopamine is more than a name, it's a promise to empower the next generation of medical minds. With a mission to make medical education accessible, affordable, and revolutionary, Dopamine is here to redefine learning for undergraduates. Join us in shaping the future of healthcare, one brilliant mind at a time. What if education wasn't just about learning? What if it was about igniting a lifelong passion for excellence? Dopamine is more than a name, it's a promise to empower the next generation of medical minds. With a mission to make medical education accessible, affordable, and revolutionary, Dopamine is here to redefine learning for undergraduates. Join us in shaping the future of healthcare, one brilliant mind at a time. What if education wasn't just about learning? What if it was about igniting a lifelong passion for excellence? Dopamine is more than a name, it's a promise to empower the next generation of medical minds. With a mission to make medical education accessible, affordable, and revolutionary, Dopamine is here to redefine learning for undergraduates. Join us in shaping the future of healthcare, one brilliant mind at a time. What if education wasn't just about learning? What if it was about igniting a lifelong passion for excellence? Dopamine is more than a name, it's a promise to empower the next generation of medical minds. With a mission to make medical education accessible, affordable, and revolutionary, Dopamine is here to redefine learning for undergraduates. Join us in shaping the future of healthcare, one brilliant mind at a time. What if education wasn't just about learning? What if it was about igniting a lifelong passion for excellence? Dopamine is more than a name, it's a promise to empower the next generation of medical minds. With a mission to make medical education accessible, affordable, and revolutionary, Dopamine is here to redefine learning for undergraduates. Join us in shaping the future of healthcare, one brilliant mind at a time. What if education wasn't just about learning? What if it was about igniting a lifelong passion for excellence? Dopamine is more than a name, it's a promise to empower the next generation of medical minds. With a mission to make medical education accessible, affordable, and revolutionary, Dopamine is here to redefine learning for undergraduates. Join us in shaping the future of healthcare, one brilliant mind at a time. What if education wasn't just about learning? What if it was about igniting a lifelong passion for excellence? Dopamine is more than a name, it's a promise to empower the next generation of medical minds. With a mission to make medical education accessible, affordable, and revolutionary, Dopamine is here to redefine learning for undergraduates. Join us in shaping the future of healthcare, one brilliant mind at a time. What if education wasn't just about learning? What if it was about igniting a lifelong passion for excellence? Dopamine is more than a name, it's a promise to empower the next generation of medical minds. With a mission to make medical education accessible, affordable, and revolutionary, Dopamine is here to redefine learning for undergraduates. Join us in shaping the future of healthcare, one brilliant mind at a time. What if education wasn't just about learning? What if it was about igniting a lifelong passion for excellence? Dopamine is more than a name, it's a promise to empower the next generation of medical minds. With a mission to make medical education accessible, affordable, and revolutionary, Dopamine is here to redefine learning for undergraduates. Join us in shaping the future of healthcare, one brilliant mind at a time. What if education wasn't just about learning? What if it was about igniting a lifelong passion for excellence? Dopamine is more than a name, it's a promise to empower the next generation of medical minds. With a mission to make medical education accessible, affordable, and revolutionary, Dopamine is here to redefine learning for undergraduates. Join us in shaping the future of healthcare, one brilliant mind at a time. What if education wasn't just about learning? What if it was about igniting a lifelong passion for excellence? Dopamine is more than a name, it's a promise to empower the next generation of medical minds. With a mission to make medical education accessible, affordable, and revolutionary, Dopamine is here to redefine learning for undergraduates. Join us in shaping the future of healthcare, one brilliant mind at a time. What if education wasn't just about learning? What if it was about igniting a lifelong passion for excellence? Dopamine is more than a name, it's a promise to empower the next generation of medical minds. With a mission to make medical education accessible, affordable, and revolutionary, Dopamine is here to redefine learning for undergraduates. Join us in shaping the future of healthcare, one brilliant mind at a time. What if education wasn't just about learning? What if it was about igniting a lifelong passion for excellence? Dopamine is more than a name, it's a promise to empower the next generation of medical minds. With a mission to make medical education accessible, affordable, and revolutionary, Dopamine is here to redefine learning for undergraduates. Join us in shaping the future of healthcare, one brilliant mind at a time. What if education wasn't just about learning? What if it was about igniting a lifelong passion for excellence? Dopamine is more than a name. It's a promise to empower the next generation of medical minds. With a mission to make medical education accessible, affordable, and revolutionary, Dopamine is here to redefine learning for undergraduates. Join us in shaping the future of healthcare, one brilliant mind at a time. What if education wasn't just about learning? What if it was about igniting a lifelong passion for excellence?
இப்போ திருப்பி ஃபர்ஸ்ட்ல இருந்து ஸ்டார்ட் பண்ணட்டுமா சார்? ஃபர்ஸ்ட்ல இருந்து ஸ்டார்ட் பண்ணட்டும். எப்போ நீங்க எனக்கு தம்ஸ் அப் காட்டுவீங்களா இல்ல? Hello everyone, good morning. I am Dr. S.Kanimozhi, your course director for Physiology here at Dopamine. As future medical practitioners, in your clinical practice, you are going to encounter a lot of patients with gastrointestinal disorders, be it a simple gastritis or to a complex gastrointestinal diseases. The topic of discussion today is going to lay the foundation for your understanding of the disease and also the basis for the treatment of these disorders. So without wasting much delay, let us dive into today's topic. Under the competency number 4.6, we are going to discuss salivary secretions and gastric secretions. Okay. So what are the objectives for today's class? I already told you we are going to look at the salivary secretions and gastric secretion. So before going into the details of salivary and gastric secretion, we need to know a brief understanding of the physiological anatomy of the GI tract. So the first few minutes will be dealt with the physiological anatomy of the GI tract, following which we will be looking into detail about the salivary glands. From an exam point of view, what are the important topics that a student is expected to know? As a first year MBBS graduate, you should be able to name the major and minor salivary glands, the type of salivary secretions that comes from these glands, the composition and functions of saliva, the mechanism of salivary secretions, and the control mechanisms that play in the salivary secretions. And finally, you should be able to list out the abnormalities related to salivary secretion. So all these topics can individually come as a short note. The clinical questions can come as a clinical reasoning questions, as well as everything put together can also come as a long essay question. Okay, so now when we are talking about the digestive system, we all know what is the most important function of the digestive system. It does the process of digestion and absorption. So the most important function of our digestive system is digestion. When I talk about digestion, it includes both the mechanical digestion and the chemical digestion. Mechanical digestion is breaking down of the larger food macromolecules into smaller process, and the chemical process of digestion is the process where the different enzymes act upon these mechanically digested food particles and convert them into absorbable simpler compounds. So that is the first part of the digestive system. So the first function of the digestive system is the breakdown process. Okay, so once it is broken down into simpler absorbable substance, what is going to happen? Then the process of absorption is going to happen. Okay, so the main aim of our digestive system is to digest and absorb the nutrients. At the same time, once the nutrients are all absorbed from the digestive material, what is remaining? The waste process. So the elimination of the waste process, elimination of waste products via feces is also a part of the function of the digestive system. So to help in this process of breakdown, absorption and elimination of waste substances, the gastrointestinal system helps. So what are all the components of the gastrointestinal system that helps in the process? So the GIT or the gastrointestinal tract consists of the mouth, the esophagus, the stomach, intestine and the accessory organs. So what is it? I will repeat. Our gastrointestinal tract consists of the mouth. The gastrointestinal tract consists of the mouth, the esophagus, the stomach, the small intestine, large intestine, rectum, anus, as well as the accessory organs that helps in digestion. So when we are talking about the structure of the GI tract, we can broadly classify it into two. One is the alimentary tract or the gastrointestinal tract. So when I'm saying alimentary tract or the gastrointestinal tract, it comprises of a continuous tube. So this continuous tube is in constant exposure to the external environment. So this continuous tube starts with the mouth. The continuous tube starts from the mouth and ends in the anus, and it runs through the thoraco-abdominopelvic cavities. So what does this alimentary tract or the gastrointestinal tube is made up of? It is made up of, it starts from the mouth. So it consists of the mouth, it consists of the mouth, the pharynx, the esophagus, the stomach, the small intestine and the large intestine. So basically the alimentary canal consists of mouth, pharynx, esophagus, stomach, small intestine and large intestine. And if you look at the length of the GI tract, it is around 5 to 7 meters in a living person. Okay. So the length of the GI tract is around 5 to 7 meters in a living person, and in a cadaver it is around 7 to 9 meters. Now moving on to that was our alimentary tract. Now moving on to the accessory digestive organs. What are the accessory digestive organs? So you can see the entire list of accessory digestive organs that has been listed here. This includes the teeth, the tongue. It includes the teeth, tongue, the liver, the gallbladder, pancreas and the salivary glands. So what is the use of our teeth? We all know it helps in grinding, chewing, breaking down the food particles. So what is the most important function of the teeth? It aids in the physical breakdown of the food. Tongue assists in chewing, swallowing and mixing of this food. Now coming to the glandular accessory organs. So these glandular accessory organs are mainly the salivary glands which secretes saliva, the liver which secretes bile, the gallbladder which stores this bile, the pancreas which produce various pancreatic enzymes. Okay, so the accessory organs are mainly the teeth, the tongue and various glandular structures like the salivary gland, the liver, the gallbladder and the pancreas. So that is again a picture showing you the summary of the gastrointestinal tract. As I already told you, the gastrointestinal tract could be divided into two. The alimentary tract, which is basically starting from the mouth, going through the pharynx, then the esophagus. So it starts from the mouth, then it goes to the pharynx, then we have the esophagus, then we have the stomach. From the stomach we have the small intestine which is made up of duodenum, jejunum and ileum, and then we have the large intestine which is made up of the colon, basically the ascending colon, transverse colon, the descending colon, sigmoid colon, the rectum and the anus. And then these are all the parts of the alimentary tract. Then we have the accessory organs. Accessory organs are the ones which helps in the process of digestion and absorption, which basically consist of our teeth, the tongue and glandular structures like the salivary gland, the pancreas, the liver, the gallbladder. Okay, so that is the different parts of the GI tract. Now in this GI tract, as I have mentioned here, we have around 6 different sphincters. Okay, so what are sphincters? Basically circular muscles which is present at the either opening or ending of a particular region of our GI tract. Sphincters are circular muscles present at the opening or ending of a particular GI segment. Okay, so here let us start with the upper esophageal sphincter. So we have the upper esophageal sphincter, then we have the lower esophageal sphincter, then we have the pyloric sphincter, we have the ileocecal sphincter, then we have the internal and external anal sphincters. So how many sphincters are there in our GI tract? There are around 6 sphincters. So now moving on to the functions of the digestive system. What is the overall function of the digestive system? Already I told you, in a broad sense, it helps in the process of mechanical and chemical breakdown of food, and then once it is broken down, absorption of the food particles. Once the food particles are absorbed, it helps in the elimination of the undigested and unwanted materials through feces, right? So we will start with the first most important function, that is injection. So the first most important function of our GI tract is ingestion, that is taking in of food into the mouth. Second is once the food enters into the mouth, what is going to happen? Not only the mouth, from there once it enters into the different parts of the GI tract, namely the stomach and the small intestine, what is going to happen? Various substances that is going to aid in the digestive process are going to be secreted, namely water, acid, buffers and different enzymes are secreted into the gastrointestinal tract. So second function is, following ingestion, the digestive system is going to do the function of secretion. Once these enzymes are all secreted, then what is going to happen? The food particles, which are larger macromolecules, need to be broken down into smaller particles. That is called as churning of food, okay, which is happening mainly in the stomach. Then once it is churned, it has to be mixed. It has to be mixed with the secretions. Only when the food particles that are broken down are mixed with the secretion, the digestive enzymes that are present in the secretion is going to properly act on them. So after grinding the food, it gets mixed with the digestive secretion. Once it is acted upon by these digestive enzymes, then what needs to be done? It has to move from one segment of the GI tract to the next segment. Then only one process is completed, the next process will happen. For example, the grinding and churning, once it is completed in the stomach, the food will be propelled into the small intestine. Okay, so that is the other function. So mixing and propulsion of food. So once it is going into the small intestine, what will happen? Once the churned and mixed food, now we don't call it any more food, we call it as chyme. After it is churned and mixed with the gastric juice, we call it as chyme. So this chyme will then move into the next segment, as I told you, which is the next segment, the small intestine, right? So once it moves into the small intestine, what is going to happen? Further breakdown and absorption of this digested food particle will happen. So the next function is digestion and absorption. Okay, so after where it is getting absorbed, so the complex macromolecules that was present in our food has been acted upon, it has been mechanically and chemically digested, and it has been absorbed. Once it is absorbed from the intestinal lumen, where it will go? It will go either into the bloodstream or into the lymph. Most of the products that are being most of the nutrients that are absorbed into the blood, and it is the fat which is absorbed into the lymph. The rest of the nutrients are absorbed into the bloodstream. Okay, so absorption is also over. Finally, whatever is remaining, the toxic unwanted substances that is present in the food, okay, and undigested food materials, the fibers, everything is defecated out of the GI tract from through the anus in the form of feces. So this summarizes broadly all the functions of the digestive system. So I will repeat it again. Digestive functions involve, you can instead of simply memorizing it, think what is going to happen. We are going to take food into the mouth, so that is ingestion. Once the food enters into the mouth, it has to be acted upon by various digestive enzymes. So the secretion of different digestive enzymes and water, acids and other buffers. Once these are secreted, then what will be the third function? Mixing and propulsion. Following mixing and propulsion, digestion, absorption and finally defecation. Okay, so that summarizes the functions of our digestive system. This can come as a Viva question. Now, to move into our actual topic of salivary secretion and gastric secretion, we need to understand the histology of the alimentary tract. This is very, very important. Okay, all those functions what I previously mentioned, how the GI tract is able to achieve these functions. By looking at these histology of the alimentary tract, we will be able to know what are all the different structures that is helping to achieve those functions. So broadly, the entire GI tract has 4 layers. So the gastrointestinal tract wall is basically commonly made up of 4 layers. So what are those 4 layers? From inside to outside. So when we look at the GI tract wall, it is made up of 4 layers starting from our esophagus until the colon. These layers are going to remain the same. So the innermost layer, when we are looking from, I have enumerated here from inside to outside. So the first layer from inside to outside will be the mucosa layer. Then we have the submucosa. Then we have the muscularis externa. Then we have the serosa or So here are the 4 layers of our alimentary tract. Starting from inside to outside, as I have already mentioned, the first layer is a mucosa layer. Then we have the submucosa layer. Following the submucosa, you have the muscularis externa, and the outermost layer is the serosa or adventitia. Okay. So here is a detailed picture which is showing you those layers. I want you to first look at the mucosa layer. So what did I say? The mucosa layer is the first layer, innermost layer. So if you look at the mucosa, it is not a single layer. This mucosa is made up of mucosal epithelial cells. Then next to the mucosal epithelial cells, we have the layer of lamina propria, and the last layer of the mucosa is the muscularis mucosa. Okay, mucosa is actually this mucosa layer. So this is the mucosa layer. This mucosa layer is actually having 3 different subunits: muscularis, the mucosal epithelium, then lamina propria, then the muscularis mucosa. Next to the mucosa layer is the second layer, that is the submucosa. So what you can see is the submucosa layer. Now if you look at the submucosa layer, this is also the following the submucosa layer, you have the muscularis layer. Okay, so this muscularis layer is again having 2 different types of arrangement of the muscle. The inner muscular layer is made up of a circular muscle. Then the next layer, outer, is made up of a longitudinal muscle. What type of muscle are these? Smooth muscles. These are the smooth muscles of the GI tract. Okay, then after that, the last layer is your, as I told you, the serosa. The last layer is the serosa. Okay, then what is this written as enteric plexus? What do we mean by enteric nerve plexus? The enteric nerve plexus are nothing but the intrinsic nervous system that is present inherently in the GI tract. Okay, because of the presence of this enteric nervous system only, even when the GI tract is cut from the external nerve supply, that is from the autonomic nerves, still it is able to function properly because of its own enteric nervous system. This enteric nervous system is basically made up of 2 types of nerve plexus. One is called a submucosal nerve plexus, the other is called as myenteric nerve plexus. Now as the name indicates, you can understand the submucosal. Where do you expect to see the submucosal nerve plexus? As the name indicates, the submucosal nerve plexus is located in the submucosal layer. Okay, then the myenteric nerve plexus is located between this circular and longitudinal muscle. Between this is the inner circular muscle, this is the outer longitudinal muscle. Okay, so between these 2 muscle layer, you have another nerve plexus which is called as the auribax or myenteric nerve plexus. I will be shortly telling you the functions of these nerve plexus. Okay, so now I think you have a brief idea of the histology of the alimentary canal. Now to draw this diagram in your exams is going to be very, very time consuming, and also reproducing this picture in your examination is difficult. This was shown here for your better understanding. So I would suggest when you are writing about the histology of the GI tract or the functional anatomy of the GI wall, I prefer you would draw a diagram like this. Okay, this is a simplified version of the same. So you have the mucosa layer. The mucosa layer is made up of 3: epithelium, lamina propria, and muscularis mucosa. Then we have the submucosa, muscularis layer, and the adventitia or serosa. Okay, so let us look at the functions of each of these layers. So as I told, mucosa means how many layers should you remember? You have to remember 3 layers. The first is the epithelium. Okay, so now as I already told you, starting from the mucosa till the serosa, the layers are going to be almost similar from esophagus till the colon. But slight variations will happen here and there, and it is those slight variations which you are supposed to remember. When it comes to the mouth, the pharynx, esophagus and the anal canal, the mouth, pharynx and esophagus, and then finally to the anal canal. The type of epithelium is non-keratinized stratified squamous epithelium. Whereas when it comes to the stomach and intestine, it is a simple columnar epithelium. Okay, why is this epithelium different? Because of the functional capability. Because stomach and intestine are the major areas of our GI tract where maximum digestion and in small intestine absorption of these nutrients is happening. So to aid for that function, a simple columnar epithelium is more convenient. Okay. And this epithelium is not only made up of these type of epithelial cells, it is also studded with cells called as enteroendocrine cells. So in the mucosa layer, we have certain specialized cell called as enteroendocrine cells. Why are we calling them as enteroendocrine cells? Because these are the endocrine cells that is present in the enteric nervous system. That is why enteroendocrine. Enteroendocrine cells means the endocrine cells that are present in the enteric nervous system. So what is the basic function of any endocrine cell? To secrete hormones. The same way, the enteroendocrine cells are the endocrine cells that is present in the epithelium of your GI tract that secretes various GI hormones. So secretion that comes from these enteroendocrine cells are called as GI hormones. In our discussion on GI hormones, we have discussed in detail about all these things. Okay. Now, so that was the epithelial layer of the mucosa. Following the epithelial layer, so we have the surface epithelial layer. Then we have the lamina propria. So as you can see in this picture, this is a connective tissue. Lamina propria is basically made up of connective tissue which has a lot of blood vessels and lymphatic. What I want you to remember here when it comes to lamina propria is it contains something called as malt. So the lamina propria contains malt. Please remember malt. What does malt stand for? Mucosa associated lymphoid tissue or mucosa associated lymphatic tissue. Okay, so what is the function of this? As I already told you, our alimentary tract is a continuous tube which is in direct contact with the external environment through the mouth as well as through the anus. So it is highly prone for infection from outside. This malt helps us from preventing these infection to enter into the GI tract. Okay, then the last layer of our mucosa is the muscularis mucosa. What is the last layer of our mucosa? Here you can see the last layer of the mucosa is the muscularis mucosa. Mucosa is made up of epithelium, lamina propria, which is a connective tissue rich in blood vessels and lymphatic blood vessels and lymphoid tissue, and then finally the third is our muscularis mucosa. Please don't confuse this muscularis mucosa with the muscularis externa, okay, or muscularis propria. Okay, what you see here is the muscularis externa or muscularis propria, and what is present in your mucosa is called as muscularis mucosa. Okay, so what is the function of this muscularis mucosa? You look at this mucosa. How is this mucosa? Is it smooth or it is thrown into folds like this? Yes, you're right. It is thrown into multiple folds. These multiple folds are present because of the contraction of this muscularis mucosa. Because of the contraction of the muscularis mucosa, the GI tract is not a uniform structure. It is thrown into several folds. Now, what is the importance or what is the physiological significance of these folds? The physiological significance of these folds is nothing but it increases the surface area for digestion and absorption. It increases the surface area for digestion and absorption. Okay. That is about our, which layer did we see? We saw about the mucosa layer. Now, from the mucosa layer, we are moving on to the submucosa. So, one important take-home message which I want you to remember here is the malt, that is mucosa associated lymphatic tissue, which is present in the lamina propria. Very important, they can ask you in your multiple choice question. Then the muscularis mucosa. Muscularis mucosa is different from the muscularis externa. The muscularis mucosa that is present in your mucosa layer is, the main function is, it is responsible for the folding that is present in the mucosa layer, and this foldings is increasing the surface area for absorption and digestion. Now we are moving on to the second layer, that is the submucosa layer. Okay, submucosa layer, similar to your lamina propria, is an areolar connective tissue. Okay, so when I say connective tissue, what is it going to do? It is going to bind the mucosal layer with the muscularis layer. Okay, that means it is binding the first layer with the third layer. That is the function of the submucosa. Additionally, it also contains glands, blood vessels and lymphatic tissue that receive the absorbed food material molecules. Another very important thing I want you to remember, when the moment you hear the word submucosa, what I want you to remember is submucosal plexus. Okay, that is the most important thing you have to remember. In the submucosa, a part of the enteric nervous system is there. And what is that called as submucosal plexus? Another name for the submucosal plexus is also called as Merson's plexus. Merson's plexus. Okay. So, what submucosa starts with what? Yes. Okay. So, please remember secretion. In GI system, to aid in the function of digestion and absorption, the GI system has to do two important things. One is secretion of enzymes and other substances, then moving the substances from one region to the other, right? This is done with the help of the enteric nervous system. Basically, enteric nervous system is helping for secretion and GI motility. So, the Merson's plexus or the submucosal plexus, on stimulation, they are going to increase the secretions in the GI tract. Okay, please remember only two key points. The submucosa, in the submucosa lies the submucosal nerve plexus. What are the submucosal nerve plexus? These are part of the enteric nervous system. Okay, now this submucosal nerve plexus is also called as Merson's nerve plexus. And what is the function? On stimulation, submucosal nerve plexus causes the secretion of different GI, secretions from different glandular tissues in the gastrointestinal tract. Okay, submucosa secretion. Remember S for S. Okay. Now let us move on to the third layer, that is the muscularis layer. Okay, so we have the third layer, that is the muscularis layer. This muscularis layer, usually, already at the starting I told you, the muscularis layer is made up of smooth muscle, GI smooth muscle, which is inner circular and outer longitudinal muscles. Okay, so we have the inner circular muscles and outer longitudinal muscles. In between these layers of muscles, you have the second nerve plexus, that is called as the myenteric nerve plexus. This myenteric nerve plexus is also called as Auerbach's nerve plexus. Myenteric nerve plexus is also called as Auerbach's nerve plexus. Okay, so where are they located? The second part of the enteric nervous system, that is the myenteric nerve plexus, is located in between the circular muscle layer and the outer longitudinal muscle layer. Since it is located in the muscular layer, remember M for M. So, myenteric plexus is located in the muscular layer. Okay, and what is it going to cause? Motility of GIT. Okay, myenteric plexus is going to cause the motility of GIT. Okay, now a short question. What about the Merson's plexus? Where are they located? Yes, you're right. The Merson's plexus are located in the submucosal region. Submucosa, yes. So it is going to cause secretion from the glandular tissues in the GI tract. Okay, so that is the most important part which I want you to remember. Additionally, also remember, in the muscularis layer, it is predominantly only smooth muscle, but there are certain areas where you can also find skeletal muscle. Predominantly it is smooth muscle, but there are regions in the third layer, that is the muscularis layer, you can also find certain skeletal muscle. Okay, where do you see? Especially in the mouth, the pharynx, superior and middle part of the esophagus. Okay, here you see skeletal muscle. What is the function? Why should skeletal muscle be here? Skeletal muscle, because for the voluntary swallowing. These skeletal muscles are for the act of swallowing. Then another place where you see skeletal muscle is the external anal sphincter. Two places. One is for the swallowing. So muscles of swallowing, muscles of the mastication, there you find the skeletal muscle. The second place where you find skeletal muscle is in the external anal sphincter. Okay, what is the function of this external anal sphincter? This permits the voluntary control of defecation. When we are seeing defecation reflex, we will look at its function in detail. Okay, so that is the muscularis layer. Why it is important? But please don't forget, it is predominantly made up of smooth muscle, lying in between the layers of the smooth muscle is your Auerbach's plexus or the myenteric plexus, stimulation of which causes motility of the GI tract. Other than that, there are certain regions where skeletal muscle is present. This is important for your MCQs. And also sometimes they can ask you in Viva, what are those special areas where you can find skeletal muscle in the muscles of mastication, and where it helps in the voluntary phase of swallowing, and also in the external anal sphincter skeletal muscle is present. Okay. Now let us move on to the last layer, that is the outermost layer, which is the serosa. Serosa is also again a alveolar connective tissue which provides the structural support to the GI tract. Okay, except for the esophagus. In esophagus you don't have serosa. Esophagus, this is an important point again for MCQ. Esophagus lacks serosa. Instead of it, there is a single layer of connective tissue called adventitia. Okay, so in case of esophagus, we don't call it, the connective tissue is not called as serosa, the connective tissue is called as adventitia. The rest of the region, the outermost layer of connective tissue is called as serosa. Okay, so that completes our discussion on the histology of your GI tract. Now finally, when we are talking about how are all these secretions and GI motility regulated, it is with the help of nerves, right? All these functions are regulated with the help of our nerves. So this neural regulation could be, two types of neural regulation is present for the GI tract. It could be an intrinsic neural regulation and extrinsic neural regulation. Two types of neural regulation, intrinsic and extrinsic neural regulation. When I say intrinsic, from the name itself, by now you should have understood, intrinsic means it is present inherently in that particular region. So GI tract already has its own intrinsic nervous system which is called as enteric nervous system. Now you help me telling what are the two components of the intrinsic nervous system. Yes, Meissner's plexus, then myenteric plexus. Okay, so the Meissner's and myenteric plexus are the enteric nervous system. Then we also have the extrinsic control, extrinsic neural regulation. So extrinsic neural regulation is through the autonomic nerves, which forms part of it is parasympathetic nervous system, sympathetic nervous system and parasympathetic nervous system. The sympathetic and parasympathetic nervous system forms the external control of the GI neural regulation. See, this parasympathetic and sympathetic do not directly go and innervate the gland or the gastrointestinal structures. They end on the enteric nervous system. So this extrinsic nervous system actually end on the intrinsic nervous system and modulate the function of the enteric nervous system, and that brings about its action. Okay, for example, stimulation of the parasympathetic nervous system. Stimulation of the parasympathetic nervous system is going to increase the GI motility. GI motility is increased, GI secretions are increased. The opposite effect is seen in case of sympathetic nervous system. So to bring about this action, it doesn't directly go and innervate the gland, or it doesn't directly go and innervate the GI tract. It is ending on the enteric nervous system, and the enteric and modulates the function of the enteric nervous system. Okay, this is one point you have to remember. That pertains to the neural regulation of the GI tract. So regulation of GI functions by nerve is by neural regulation. Okay, now we were talking about the enteric nervous system, right? The myenteric and the Meissner's. So how does this enteric nervous system bring about its action? Through short reflexes. Please remember the name, short or local reflex. The enteric nervous system, that is the myenteric and the Meissner's nerve plexus, they bring about their action through short local reflexes. The extrinsic nervous system, they bring about their action through long reflexes. Okay, already I told you, submucosal is for secretion, the myenteric is for motility. Okay, now let us look more into little bit more about this enteric nervous system. So here you can see the myenteric plexus, submucosal plexus, which forms the enteric nervous system. When you look at this enteric nervous system, they are made up of short interneurons. They are made up of short interneurons, okay, so which is connecting the afferent and efferent neurons within the GI tract locally, okay. And the reflexes that is initiated here are called as local reflexes. If the reflexes are initiated via the extrinsic nervous system, that is the autonomic nervous system, we call it as long reflexes. Let us see a simple illustration of the long and short reflexes, because understanding these long and short reflexes is very important, because as we go through GI motility, there if you don't have an understanding of what is a short reflex, what is a long reflex, everything will look Greek and Latin. So please take some time and try to understand these short reflexes and long reflexes, okay. So what are we discussing? Basically, how the nervous system regulates the gastrointestinal functions. That is what we are going to see. Basic important gastrointestinal functions are brought about either by GI motility or through GI secretions. So how are these two functions regulated via the nerves? Through short reflexes and long reflexes. When I say short reflex, I mean the reflex is locally mediated within the enteric nervous system. When I say long reflex, then the reflex is mediated via the autonomic nervous system. Is it clear? Long reflexes means it is autonomic nervous system. Short reflexes means it is enteric nervous system. I hope these 2 points are clear. Now let us see how a short reflex is initiated. Any stimuli, okay, be it the stretching of the gastrointestinal wall, okay, which will stimulate the mechanoreceptor, or the osmotic content of the osmolarity of the content in the GI tract, that is going to stimulate the osmoreceptor, or the chemical composition, presence of proteins, lipids, fat. So the chemical composition of the food, be it stretching of the GI tract, or the chemical composition, or the electrolyte composition of the food, all this is acting as a stimuli, and this type of stimuli is going to stimulate the respective receptors, okay. So any reflex, what will it have? You need to have a stimuli. Any reflex, first thing we will have the stimuli, then we will have the receptor which receives the stimuli, then we have the afferent, then we have the center, then we have the efferent. From the center will give its input through the efferent, and finally we have the target organ, in which the response is happening, right? So these are the components of any reflex. So here the stimulus could be any stimulus, be a mechanical, chemical stimuli. Now the stimuli is going to go and stimulate its respective receptors. Once the respective receptors are stimulated, afferent impulses are initiated, and they are going to increase or decrease the function through the local enteric nervous system, okay. This local, because these receptors, once they stimulate or inhibit the auribax and myenteric plexus, the result of which is going to be either stimulation of a gland so that secretion is happening, or a stimulation of a stomach or your intestine so that contraction is happening. So any of this could be a response. When all this is happening within the GI tract without involving the brain, without involving the higher center, we call it as a short reflex. Example of a short reflex is enterogastric reflex, okay. We will see about this reflex when we are discussing GI motility. Just now, for now, you remember enterogastric reflex is an example of a short reflex. That means it is mediated, initiated, and the response is happening within the GI tract through the enteric nervous system. Now let us go to the long reflex. What did I say? Long reflex means central nervous system is involved. So be it a stretching, same stimuli, whatever I told for short reflex, or sight, smell, thought of food, okay. What is going to happen is the stimulus is going to stimulate the receptors. The receptors, afferent impulses are now going to the central nervous system. For long reflex, afferent impulses is going to the central nervous system. Then from the central nervous system acts as the center. From the central nervous system, usually afferent is through vagus, okay, afferent vagal fibers. Usually it is the afferent vagal fibers which carries it to the center in the vagal nuclei in the medulla. Mostly it is the vagal nuclei in the medulla. Okay, then from the vagal nuclei in the medulla, the efferent impulses via the efferent vagal impulses will come to the local nerve plexus. Please remember, the efferent is not directly going to the target organ. The efferent will first come to the local enteric nervous system. After coming to the local enteric nervous system, then it follows the same path as a short reflex, finally going to the effector organ. So this is how a long reflex is acting. Okay, and example of a long reflex is vagovagal reflex. Vagovagal reflex. Okay, as well as your salivary secretion, that is also an example of your long reflex. Okay, so now I hope you have an understanding of how the neural mechanism is acting. What are the different components of the neural mechanism that regulates gastrointestinal functions, and how they regulate via the short reflexes and long reflexes. Finally, the functions of our gastrointestinal tract is also influenced by hormones. Okay, so when it comes to hormones, similar to the GI tract, we have the intrinsic hormones, extrinsic hormones. Intrinsic hormones means all the hormones that is secreted by the enteroendocrine cells, that is the GI hormones. Extrinsic hormones means hormones that is coming from the other endocrine glands like the thyroxine, cortisol, etc. Okay, so that summarizes our brief discussion on the gastrointestinal physiology, gastrointestinal functional anatomy of the gastrointestinal tract. So with this knowledge, only when we have this knowledge, we will be able to understand the rest of things. Okay, so now we are very clear about the different layers, the nerve plexus, how these nerve plexus act, and all those things. Now let us move on to the salivary secretions. Okay, fine. So to understand salivary secretion, first let us look at the structure of the secretory units that is present in the saliva. Okay, the secretory units of the saliva is called as, the secretory unit of the saliva is, what is it called as? Salivon. Okay, this is an important MCQ. What is the name of the secretory unit that is present in the saliva? The secretory unit of saliva is called as salivon. This secretory unit is basically made up of the acini and the intercalated duct. The acini and the intercalated together form the salivon. Okay, so now when you look at this picture, what you can see, the salivary gland is made up of two structures. One is the acinus, the other is the duct. Okay, so the salivary gland is made up of acinus and the duct. So here you can see the ductal system, here you can see the acinus. When I am talking about acinus also, you have the serous acinus and then you have the mucous acinus cell. So when you look at the alveoli, when you look at the structure of the alveoli of a salivary gland is similar to the alveoli of our lungs. Okay, so we have the alveoli which is having two types of cells, serous cell, mucous cell. Okay, in the center you have the cavity called as acinus. The secretions from the serous cells and mucous cells will be emptied into this acini. Okay, and this central cavity of the acini is continuous. The central cavity of this acini is continuous as the duct. So the secretion from the serous and mucous cell will be emptied into the central acini. From there the secretions will be carried via the ducts. Before going into the duct, let's discuss about the serous and mucous cells. The serous cell contains zymogen granules which contains the enzymes. The digestive enzymes of saliva. Okay, can you name the digestive enzymes of saliva? Yes, the ptyalin or amylase, the lipase, all these digestive enzymes of saliva is present within the zymogen granules that is formed in the serous cell. Okay, other than that, from the name itself, what does it say? Serous. Serous means what? It is going to produce secretions that is watery. Okay, so when the serous cells of the salivary glands are stimulated, it is going to produce secretions that is rich in water. Along with it, it will also produce the secretion is also going to contain the digestive enzymes. Now mucus, as the name indicates, what is going to be present in the mucus? The mucus cells are going to be producing secretion that is rich in mucus, that is thick and viscous. Okay, the secretion that is coming from the mucous cells are thick, viscous and rich in mucin. Okay, now the secretions are emptied into the central acini. From there the ducts are carrying it. So if you look at the hierarchy of the duct also, from the acini you have the duct is called as intercalated duct. The intercalated duct will join, many intercalated duct is joining to form the intralobular duct. Intercalated ducts join to form interlobular ducts. Intralobular ducts join to form the interlobular ducts, and interlobular ducts join to form the main the main salivary duct. Okay, so that is the structural arrangement of the salivary gland. Okay, so to recall, what is a salivon? The structural and functional unit of salivary gland is called a salivon. It is basically made up of the acini and the intercalated duct. Only the acini and intercalated duct form the salivon. Okay, and if you look at the arrangement from this picture, you can see this type of arrangement, this type of structures with alveoli and ductal system are called as racemose type of arrangement. Racemose means bunch of grapes. Okay, so that was briefly the structure of the salivary glands. Now coming to the classification of salivary glands. How are we going to classify salivary glands? Salivary glands can be classified based on their size and location, and also based on the type of secretion that comes from the salivary glands. So first let us see the based on the size and location, how are these salivary glands classified. So we have major salivary glands. We have three major salivary glands. What are those? Parotid glands, submandibular gland, sublingual gland. I will repeat it again. The major salivary glands are the parotid gland, submandibular gland and sublingual gland. Then we have the minor salivary glands like the lingual mucous salivary gland, linguoserous salivary gland, buccal glands, labial glands and palatal glands. Depending upon where they open and bring about their secretion, empty their secretion, those names are given to these minor salivary glands. Okay, same thing applies to the major salivary glands also. Okay, so now let us look in detail about these major salivary glands, which is the first and most important. The largest salivary gland is the parotid gland. So the parotid gland is the largest salivary gland. It weighs around 14 to 28 grams. They are located bilaterally. So we have a pair of salivary glands on both the sides. So the weight of the parotid gland, which is the largest salivary gland, is around 14 to 28 grams, and it provides, this is very important, 60 to 65% of the total salivary volume. Later on I will tell you whether this is at rest or when the salivary glands are active. That is also important. For now, remember 60 to 65% of the total salivary volume is because of our parotid gland. Where is this parotid gland located? Situated at the side. As you can see, the parotid gland is situated at the side of your face. Okay, just below and in front of your ear. As you can see, just below and in front of the ear we have the salivary gland. Now the secretion from the salivary gland, this is a salivary gland. Okay, this is a gland. The secretion from this gland is poured into the oral cavity. Secretions from any of the salivary glands is going to enter into the oral cavity, right? In this, then only it forms saliva. So now, in case of your parotid gland, the duct, which most important duct which carries the salivary secretion and emptied into the oral cavity, the name of the duct is called as Stenson's duct. Important MCQ question. Stenson's duct is a major parotid duct. Okay, and what does it do? It is around 35 to 40 millimeter long. The secretions from the parotid gland is emptied into the mouth through the Stenson's duct, and this Stenson's duct opens at the level of second molar teeth. Second molar tooth. At the level of the second molar tooth only, the Stenson's duct is opening. Okay, and the secretions that comes from the parotid gland is usually serous secretion. Okay, that was about the parotid gland. Now we are going to the submandibular gland. The submandibular gland is also called as a submaxillary gland. It is an irregular wall-shaped gland. 20 to 30% of the total salivary volume is because of the submandibular gland, and it weighs around 10 to 15 gram, where it is located in the submandibular triangle. And the secretions from the submandibular gland, so here you can see the submandibular gland. The secretions from the submandibular gland is emptied via the duct of Warthin. So please remember the name of the duct. For parotid gland, the secretions are brought and emptied into the oral cavity at the level of second molar tooth by the, what is the name of the duct? Stenson's duct. Now for the submandibular gland, what is the name of the duct? Warthen's duct. Okay. Finally, we have the last major salivary gland, that is a sublingual gland. This is the smallest of all three, weighs only 3 to 4 grams, and it contributes only 2 to 5% of the total volume. Where does it lie? It lies beneath the floor of the mouth, above the mylohyoid muscle. Okay. So that is your sublingual gland. Okay. Now the secretions from the sublingual gland is emptied into the oral cavity by a duct called as duct of Redefining. Please remember, for the sublingual gland, the name of the duct that carries the secretion is duct of Redefining. Okay. So that is with regards to the major salivary glands. The minor salivary glands, already I listed you the important minor salivary glands. They are usually located beneath the epithelium in almost all parts of the oral cavity. Around 600 to 1000 minor salivary glands are present, and they are classified based upon their anatomical location, as we already saw. Now, that was the classification of salivary glands based on their structure and location, major and minor. Please remember the major. What are the major salivary glands? The parotid, submandibular or submaxillary, and the sublingual parotid glands. Sublingual salivary glands. In these salivary glands, what I want you to remember is the name of the duct which brings their secretions. Okay. For each of the salivary glands, we have different ducts. Okay. Now moving on to the classification based on the properties of the secretions that is coming out from these salivary glands. So serous glands, mucous glands, mixed glands. Based on this, we have three types of glands: serous glands, mucous gland, mixed gland. Already we saw the secretions from the parotid is mainly serous. When I say serous, that means it is thin, watery, and rich in enzymes. Okay. What are the examples? Parotid, lingual. Both are, example of serous glands. Then we have the mucous glands. Mucous glands are made up of mucous cell, so they are going to produce secretions that is thick and viscous. Okay. Example, the lingual mucus, that is a minor salivary gland. Lingual mucus, buccal and palatal glands, the secretions are all mucus secretion. Then mixed salivary glands means both it has serous cell and mucus cell. Example, submandibular, sublingual. Okay. See, among the three major salivary glands, parotid is serous. Please remember, this is again important. The secretions from parotid is serous. Okay. The secretion from the submandibular and sublingual glands are mixed. Okay. So that was the classification of the salivary glands. Now we will shortly look at the innervation of salivary glands, the nerve supply of salivary glands. So the salivary glands are under the autonomic nervous system control. Okay. So the moment when I say it is under the autonomic control, we have the parasympathetic and sympathetic. Generally in GI tract, parasympathetic means increases the secretion, sympathetic means decreases the secretion. Okay. Remember this. Now when it comes to salivary gland, how is the parasympathetic supply? The parasympathetic supply originates from the superior and inferior salivary nucleus, which is located in the medulla. Okay. From the inferior salivary nucleus via the 9th cranial nerve, that is the glossopharyngeal nerve, from the inferior salivary nucleus originates your parasympathetic. Okay. Where are these salivary nuclei present? In the medulla. So in the medulla, from the inferior salivary nuclei, the 9th cranial nerve, that is the glossopharyngeal nerve, carries the preganglionic parasympathetic fibers and synapses with the otic ganglion. Okay. Then postganglionic parasympathetic. So this is preganglionic parasympathetic fiber. Now from the otic ganglion, the postganglionic parasympathetic fibers will go and innervate the parotid gland. Okay, please remember, again very important Viva and MCQ question. The parasympathetic innervation for the parotid gland is via which nerve? The glossopharyngeal nerve, that is the 9th cranial nerve. Okay, through which ganglion? The otic ganglion. Now, from the same medulla, but from the superior salivary nuclei, the origin is from the superior salivary. This is the origin. Origin is from the superior salivary nucleus. The facial nerve, that is the 7th cranial nerve. The facial nerve, through the facial nerve, the preganglionic parasympathetic fibers will end on the synapse with the submandibular ganglion. The submandibular ganglion will then give rise to post-ganglionic parasympathetic fibers to the submandibular and sublingual gland. That means the parasympathetic supply to the submandibular and sublingual salivary glands are through the 7th cranial nerve, that is the facial nerve. And for the parotid gland, it is through the 9th cranial nerve, which is the glossopharyngeal nerve. That is with regards to parasympathetic. As I already told you, when there is parasympathetic stimulation to the salivary glands, we are going to have a flow, high flow rate. Why it is high flow rate? Because it is serous, it is watery, and also low protein saliva. Because it is watery, it is low in protein but enzyme rich. Please remember this word, enzyme rich secretion, digestive enzyme rich secretion. Now looking at the parasympathetic supply. The parasympathetic supply starts from the sympathetic salivary centers, which are located in the upper thoracic segments of the spinal cord. So originates from the upper thoracic segment. The preganglionic sympathetic fibers are going to go and synapse with the superior cervical ganglion. From the superior cervical ganglion, the postganglionic sympathetic fibers are going to innervate all these major salivary glands. On sympathetic stimulation, how is the salivary secretion going to be? The salivary secretions is going to be low, it is going to be thick, it is going to be viscous, it is going to be rich in protein. Okay. Rather than, when it comes to salivary secretion, rather than telling parasympathetic stimulation increases the salivary secretion, sympathetic stimulation decreases salivary secretion, that is not the right way to actually describe. When it comes to salivary glands, what we have to tell is parasympathetic is going to produce salivary secretion that is thick, that is thin, watery, rich in enzymes. Sympathetic salivation is going to produce salivary secretions that are thick, viscous, high in protein. Okay. Now coming to the salivary blood flow. If you look at the rate of salivary secretion, it is around 50 ml per 100 milligram. Sorry, if you look at the salivary blood flow, not the rate of secretion, but if you look at the salivary blood flow, it is around 50 ml per minute of 100 grams of the salivary tissue. Okay, so this is actually very, very high flow rate, 10 times than that of other major organs. Okay, so the blood flow, it has a very high blood flow. Okay, why very high blood flow? Because salivary glands are a highly metabolically active gland. Since it is a metabolically active gland, it needs to have high rate of salivary secretion. If you want high rate of salivary secretion, then you need to have high blood flow rate per gram of the tissue, per 100 gram of the tissue. Now already we saw what will happen if parasympathetic stimulation is going to happen. So how is this parasympathetic stimulation going to produce so much of watery salivary secretion? Because of vasodilation. Okay, parasympathetic stimulation causes vasodilatation. And how does this vasodilatation occur? Via the secretion of vasoactive intestinal peptide, bradykinin and acetylcholine. Okay, sympathetic stimulation decreases the flow. So that is going to cause vasoconstriction, reducing the blood supply. Lesser blood supply, lesser rate of secretion. Okay, so that is about the rate of blood flow and the secretion. Now we are going into the stages of salivary secretion. So what did we, just to recap, what did we see so far? We saw what are the different types of salivary glands. Okay, based on their classification, what are the different types of salivary glands based on their location and the type of secretions that comes from it. Then we also saw the innervation to the salivary gland and how the blood flow to the salivary gland affects the salivary secretion. Now we are moving on to how the salivary secretion actually happens. Okay, so for that, first we will see the salivary secretion is a two-stage process. What is it? The salivary secretion, that is the secretion of saliva, is a two-stage process. First is called as the initial formation stage. The second stage is called as a modification stage. Okay, so the first stage is a primary secretion. Second stage is the modification. Okay, second is the modification stage. The primary secretion is basically happening in the acinus. The primary secretion is basically happening in the acinus, and the second modification is happening in the duct. Please remember these two things from the starting till now. When we are talking about saliva, already in the structure we saw it is made up of acinus, it is made up of duct. Okay, so when saliva is formed, it is first formed in the acinus, that is called as primary salivary secretion. Now the saliva that is formed in the acinus is modified in the duct. So that one which gets modified in the duct is called as secondary salivary secretion. Okay, so first let us look at the primary, how the primary salivary secretion is formed. Then we will move on to the second stage, that is how it is getting modified. Okay, so for the formation of saliva, that is step 1, what is needed? So these are the series of events. I have just written it down in a flow chart, so it will be easier for you to write in your exam. So first step, what happens? Stimulation of the parasympathetic nerve fibers will release the calcium that is stored in the intracellular stores. Okay, because of the release of intracellular calcium, what is happening? It is going to cause increase in the intracellular calcium concentration, is going to open chloride channel apically and potassium channel basolaterally. Opening up of the chloride channel will lead to movement of chloride into the lumen. Okay, so basically the saliva is formed and secreted into the lumen, that is the acini. Then from there it will go into the duct. Finally it will be emptied into the mouth. So now what is happening? The chloride channels are opening. Chloride ion from the acinar cell is moving into the lumen of the acini. When chloride is moving, it is an osmotically active particle. This is osmotically active. So what it is going to do? It is also going to pull sodium into it. This causes change in the osmotic pressure, which leads to movement of water into the lumen. Okay. So this is the first stage of, that was in the form of a flow chart. The same thing is depicted here in form of a picture. So first step is formation of primary salivary secretion. First step is primary salivary secretion. So for primary salivary secretion to happen, what is happening? Basically influx of calcium. Influx of calcium is going to increase the intracellular calcium concentration, so that opens chloride channels. So when the chloride channels open, what is going to happen? The chloride ions are going to come inside. When chloride is coming inside, it is also dragging sodium and water. Everything is coming inside, and it forms a saliva that is isotonic to that of plasma. It is isotonic to that of plasma. Along with these electrolytes, what is getting added? Enzymes. Okay, mainly the alpha amylase. It is also called as ptyalin. Salivary amylase or ptyalin is also getting added to the primary secretion. So the primary salivary secretion which happens in the acini is basically isotonic to that of plasma. Then here you are seeing something called as myoepithelial cells. Okay, these are the myoepithelial cells. What they are going to do? Myoepithelial cells are going to cause contraction. These myoepithelial cells basically contracts and relax. So these are all myoepithelial cells. Okay, all this blue color structures are all myoepithelial cells. So this myoepithelial cell is going to contract. When these myoepithelial cells are contracting, what is happening? The size of the acini reduces and the secretion is going to get pushed into the duct. So because of the contraction of the myoepithelial cells, the secretion that is present in the central acinus is moving towards the ductal cells. Okay, so it is moving towards the duct. So once this comes here, the second stage is starting. So once it comes into the duct, the isotonic primary salivary secretion that comes into the duct, what is going to happen? Just remember two things. Sodium and chloride is taken back. It is reabsorbed. Sodium and chloride is reabsorbed. Potassium and bicarbonate is secreted. Potassium and bicarbonate is secreted into the saliva. Sodium and chloride is reabsorbed. Okay, because of this, what is happening? The saliva becomes hypotonic to that of plasma. Because of the movement of these electrolytes, the saliva becomes now hypotonic to the plasma. So that means the end secondary salivary secretion. So this is called as secondary salivary secretion. So the primary salivary secretion is isotonic to plasma. whereas the secondary salivary secretion is hypotonic to plasma. Okay, so how the sodium and chloride is reabsorbed, I will show you here. Okay, so this is a salivary ductal cell. What we are going to see is the stage 2. Okay, secondary salivary secretion. What is happening? First, sodium and chloride are reabsorbed. How sodium? So this is a lumen. So here you have all those sodium and chloride. You want the sodium to go inside, you want the chloride to go inside. So for the transport of sodium from the lumen into the salivary ductal cell, you have the sodium hydrogen exchanger. So this sodium hydrogen exchanger takes sodium inside and pumps hydrogen ion outside. So now sodium is taken back into the salivary ductal cell. Next, what you want? You want the chloride also to go inside. So for this chloride, you have the chloride bicarbonate exchanger. Okay, so we have the chloride bicarbonate exchanger where chloride enters into the cell and bicarbonate comes into the lumen. Okay, so now bicarbonate has come out, chloride has come inside. That is why sodium and chloride are reabsorbed. Now bicarbonate is secreted into the lumen. Okay, what else needs to be secreted? Potassium also needs to be secreted. Okay, so for that what happens is, on the basolateral side you have the sodium potassium ATPase pump. So what is this pump going to do? It is going to pump sodium outside and potassium inside. So the concentration of potassium increases inside the ductal cell. Now this potassium is pumped out with the help of hydrogen potassium exchanger. Okay, so that is how potassium leaves the cell and hydrogen ion comes into the cell. Okay, the hydrogen ion which has come inside again is to maintain the pH of the cell. Hydrogen ion cannot stay inside the cell. So what is going to happen? This sodium hydrogen exchanger will again pump the hydrogen ion back outside. So this vicious cycle keeps on happening. So without confusing you, I will just summarize it again. The salivary secretion is a two-step process. Step 1 is formation of primary salivary secretion, which is isotonic to that of plasma. This happens at the level of the acinus. Okay. Along with the isotonic salivary secretion, enzymes are also getting added. Okay. Now, the acinus is surrounded by myoepithelial cells. The contraction of the myoepithelial cells, what is it going to do? The contraction of the myoepithelial cell will push the secretion that is present in the central acinus into the duct. Now, the ductal cells will act upon this secretion, the isotonic salivary secretion that has come. It will act upon it and it will do only two things. Just remember two things. It is going to take back sodium and chloride. It is going to secrete bicarbonate and potassium. Because of the alternation in this ionic composition, the saliva becomes hypotonic. So the final saliva that comes is hypotonic. Okay, is it clear? It is rich in bicarbonate and it is a hypotonic to that of plasma. Now, regulation of salivary secretion. How salivary secretion is regulated? I told you, when it comes to, that is with respect to our mechanism of formation of saliva, first step and second step. Now it comes to the regulation of salivary secretion. As we already discussed, from now on it's going to be easy because already we discussed in the general organization of GI tract. So any regulation of our GI tract is going to be through neuronal and hormonal. When it comes to salivary secretion, it is a purely reflex phenomenon. That means it is regulated only with the help of the nerves. Hormones are not involved. It is a purely reflex phenomenon. Okay. This secretion is via the salivary reflexes, which are both unconditional and conditioned reflex. Okay, when do I call it unconditional and when do I call it conditioned reflex? When do I call it as unconditioned and when do we call it as conditioned? Conditioned means previous exposure or experience should be there. Unconditioned means innate. You are born with it. Okay, that means, first let me tell you unconditioned. What do you mean by unconditioned reflex? Unconditioned reflex means the sight, the smell, the thought of food. Since you have already had it could be any food of your choice. Okay, you like a particular food, your mother is preparing it, even when you enter the house with the aroma itself, you know she is prepared this particular food, you will start salivating. That is an example of conditioned reflex. Because of your previous exposure, because you already are trained, you know that the food is going to taste very yummy, the smell is so inviting, the conditioned reflex will start. Even without the actual food into the mouth, the secretions are going to start. So that is called as conditioned reflex. Then we have the unconditioned reflex. So what do I mean by unconditioned reflex? Whether you have tasted the food or not, whether you have previous experience or not, the moment the food is present into your mouth, the moment you put the food into your mouth, the salivary secretions will start. So that is called as unconditioned reflex. So through the conditioned and unconditioned reflex, what is happening? The salivary secretions are regulated. Okay. Then as I told you, what is going to do? To the higher center. So through conditioned or unconditioned reflex, through the involvement of the higher center, the medulla is going to get stimulated. So as I already told you, the inferior and superior salivary nuclei, the reflex is going to originate here through the 9th and 7th cranial nerve, through via the otic ganglion, the 9th cranial nerve, that is a glossopharyngeal nerve, secretions are going to go into the parotid gland, and via the 7th cranial nerve through the submandibular ganglion, the submandibular and sublingual parotid glands are stimulated. Okay, as a result of which you are going to have salivary secretion that is rich in enzymes as well as water. Okay, so that completes our discussion on the salivary secretion and its regulation. Now two important things which they can ask you in your viva. Two viva concepts. One is paralytic secretion of saliva, another is augmented secretion of saliva. What do you mean? These are fancy terms. Okay, what do you mean by augmented secretion of saliva? Augmentation means what? Increasing its activity, right? So when you are stimulating the salivary glands continuously twice, the second stimulus is going to produce salivary secretion that is far much higher in amount than the first stimuli. This is called as augmented secretion of saliva. The same concept of stimulation of skeletal muscle fiber, summation, the same funda is applied here as well. Okay, then coming to the paralytic, what do we mean by paralytic secretion of saliva? Paralytic secretion of saliva is, we already saw saliva, the nerves that carry the impulses are either through the 9th cranial nerve or the 7th cranial nerve. During any surgery in the facial region, if this 9th or 7th cranial nerve, mainly the 7th cranial nerve, once it gets affected, now the parasympathetic fibers which is bringing this stimuli are getting damaged. Because of this damage, what happens? Now we will still have salivary secretions. Salivary secretions are still going to be there, but it is going to be dysregulated. There is going to be dysregulated salivary secretion. Because of this dysregulated salivary secretion, what is going to happen? The ionic composition is going to go completely disrupted, and the amount of secretion is also going to increase. So such kind of secretion is called as paralytic salivary secretion. Due to the damage to the parasympathetic nerve that carries these impulses, we are going to have a dysregulated salivary secretion, which is called as paralytic secretion of saliva. Okay. Now let us look at the properties of saliva before going into the functions of saliva. Here, normally the volume of saliva is around 1 to 1.5 liters. Okay. Approximately about 1 ml per minute of saliva is secreted. Now look at this. I want you to look here carefully. When the cell is resting, look at the flow rates. When the cell is resting, the parotid secretion is only 0.04 ml per minute, and submandibular is around 0.1 ml per minute. Overall, the parotid gland is the largest salivary gland, and when it is stimulated, the parotid gland is only responsible for 60 to 70% of the salivary secretion. That is when the gland is stimulated. But during rest, when you are not eating, okay, when the gland is at rest, you have a basal salivary output. That is mainly coming from the submandibular salivary gland. This important difference I want you to note. This is important in MCQ. When the salivary secretion, when the gland is active, parotid is the major contributor. When the gland is at rest, submandibular gland is the major contributor for salivary secretion. Okay. So it is slightly the most mixed. When the saliva is usually coming from all the three glands, it is a mixed saliva which is slightly acidic with a pH of 6.35 to 6.85. The specific gravity is around 1.002 to 1.012. The tonicity, as we already saw, it is hypotonic to that of plasma. So these are the constituents present in saliva. Like any other secretions of the GI tract, majority is water, then 0.5% is solids. Solids are again inorganic and organic. Solids are organic and inorganic. The inorganic solids are mainly gases and ions, cations and anion. The organic substances are mainly enzymes and other substances. Enzymes are basically the digestive enzymes. Okay. Other substance includes protein, blood group antigen, non-protein nitrogenous base substances that needs to be excreted out. Okay. We are more concerned with the enzymes that is present in the saliva. Now comes the functions of the saliva. First and foremost, the most important function of the saliva is preparation of the food for swallowing. Okay. So what is happening? Salivary secretion. Saliva is basically a liquid which helps in mixing of the food. So it provides an aqueous liquid medium where the food particles get mixed. Only when the food particle is getting mixed, then when the teeth is grinding them, the saliva is going to help in better grinding. Have you seen when they are when your mother is using the mixer grinder, what do? Suppose they want to make a chutney, they add coconut, then what do? They add little bit of water, then only it gets grinded better. The same thing. So saliva is giving that aqueous medium where the food is getting prepared, it is getting grinded. One, mixing is happening. Second thing, even while swallowing, the saliva adds the lubricant. The mucus in the saliva acts as a lubricant which makes it easier for the oropharynx to swallow the food bolus. Okay. So that is called as preparation of the food for swallowing. Then for appreciation of the taste also, it is very important. Saliva is important for appreciation of the taste. Why? Again, it provides a aqueous base where the food molecules are getting dissolved. Only when these food molecules are getting dissolved, they can go and stimulate the taste receptors so that we are able to appreciate the taste. Okay, another important thing what I want you to add here is saliva does not contain protein digesting enzymes. That is why when we eat a food that is rich in protein, you will have a bland taste. You will not be able to taste protein because we don't have enzymes that is digesting the proteins in the mouth. Okay. So again, digestive function, just now I told. Enzymes like ptyalin or alpha amylase digest starch, and your salivary lipase is helping to digest your fats. Okay, salivary lipase mainly is secreted from the Ebner's gland, which is a minor salivary gland. Then cleansing and protective function, very important. Cleansing, again, it is the aqueous nature of the saliva. Make sure that the food particles doesn't get hidden in different parts of your mouth. Everything is getting cleansed out. And when it comes to protective function, again, the bicarbonate that is present in the saliva helps in neutralizing the acids that is present in the food that we eat. And also IgA, saliva contains, one is bicarbonate for neutralization of the acid, then immunoglobulin A, lysozyme, okay, and then lactoferrin. All these are antibactericidal agents, so that helps us in protecting the GI tract. Okay, so much of junk food we eat from outside, they are all most of the time contaminated food, but still we don't end up having a stomach infection every time we eat street foods from outside. Why? Because of the all these IgA, immunoglobulin A, lysosome, lactoferrin that is present in saliva act as antimicrobial agents that kills the pathogenic organism that is present in the food. It helps in the role of speech also. When your mouth is dry, you will not be able to speak. Articulation is not possible in a dry mouth. So saliva, the lubricating properties of saliva also helps in phonation. Excretory function, toxic chemicals that is present in the food, heavy metals are also excreted via the saliva. It regulates body temperature. This regulation of body temperature through saliva is more developed in animals. It is more developed in animals because via panting. If you have seen animals, especially dogs and all, they put their tongue out and they keep panting. Salivary secretion keeps on coming out. Drooling of saliva happens. This drooling of saliva helps in regulating their body temperature. And saliva is also important for water balance. How do saliva bring about water balance? The thirst sensation that is present when our mouth is dry, we feel thirsty, so that helps us drink more water, so it also helps in water balance. So these are the important salient functions of the saliva. Okay, now this is just a pie chart showing the multifunctionality of saliva. When you are free, you can go through this while going through the slide, where the function and the particular chemical constituent that is present in saliva that helps in that action is given. Okay, for example, antibacterial action is mainly because of the cystatin, mucins, peroxidases that is present in the saliva. Like that, for digestion you need amylase and lipases. So all the functions of saliva with their corresponding constituent is given here. This is important for your MCQ. Okay, MCQ can be asked based on this. Now finally, before finishing our discussion on salivary secretion, we will look at the applied aspects. Okay, so we have something called as xerostomia. What is it called as? Xerostomia. Xerostomia means dry mouth. Okay, now we have a syndrome called as Sjogren's syndrome. This is a chronic autoimmune disorder which is mainly characterized by dryness. Xerostomia, xerophthalmia. Okay, it is characterized by dry eyes, which is called as xerophthalmia. Of the eyes are dry. Then xerostomia, the mouth is also dry. And dryness, rest of the body parts also. Vaginal dryness will be there, dry skin will be there, the nose is very dry, so there will be frequent nosebleed, and since the throat is dry, the patient will complain of frequent coughing. Okay, so that is Sjogren's syndrome. It's an autoimmune disorder most commonly seen in females past the age of 40 years. Next, xerostomia, just now only we discussed, that is the dryness of the mouth. Because of xerostomia, when there is decreased secretion of saliva, this is going to predispose to oral infection. Dryness of the mouth is going to predispose to oral infection. One cause of xerostomia is Sjogren's syndrome. Other common cause for xerostomia is any stressful condition. Whenever you are stressed, what is going to happen? The levels of your epinephrine is going to increase, the flight-fright reaction in all these situations. Sympathetic stimuli increases, so it decreases the salivary secretion. Then we have another term called as siluria. Siluria means what? Increased salivary secretion. Normally when there is food intake or through conditioned reflex, salivary secretion will increase. Once the process is over, it will come back to the basal level. But if it is persistently increased, we call it as siluria. Then we also have something called as silolithosis. Okay, silolithosis means stone formation in the ducts of the salivary gland. Then infection of the salivary gland, very common, mumps, seen in children. Okay, then we have dental caries. As I already told, saliva, when there is dry mouth, it is going to predispose to dental caries. Okay, because what is going to happen? The salivary flow is very important for cleansing of the oral cavity. Now that doesn't happen. Food is going to get accumulated at most of the places in and around the mouth, so that is going to be a breeding ground for bacterial infection, which is going to lead to caries. Then we have dental erosion. Again, salivary gland hypofunction is going to cause deficient remineralization of the teeth, so that leads to dental erosion. Then we have something called as gingivitis. All these are important as definitions. Okay, what is gingivitis? Due to the lack of saliva, as I already said, the food particles are present retained in the mouth, which causes infection leading to periodontitis. Gingivitis, which leads to periodontitis, inflammation of the gums. Then oral ulceration. Again, decreased salivary secretion is going to cause oral ulceration, and which can also lead to the candidial fungal infections of the mouth. Then finally, mucositis. What is mucositis? It is a painful condition where the mucous membrane of the oral cavity becomes ulcerated and inflamed due to decreased salivary secretion. Then we have another term called as dysgeusia. What is dysgeusia? Distortion of the taste sensation. Okay, as I told you, the saliva acts as a solvent, so when that is not there, we will not be able to appreciate taste sensation. Dysgeusia. Glossitis, the appearance of red, raw, dry tongue is called as glossitis. In glossitis, the piliform papillae are completely lost. Okay, so that completes our discussion on salivary secretion. Hope you understood the topic well. We'll take a short break and then we will move on to our gastric secretion. Okay, so welcome back. Now let us start our discussion on gastric secretion. Okay, so what is this yellow color liquid that is present in the stomach? Think and tell me. The gastric secretion is mainly composed of most important component in gastric secretion. What is it? It is hydrochloric acid. Okay, so other than hydrochloric acid, what are the other things that is present in gastric secretion? Pepsinogen. What else? Yes, you can help me. Think and tell. Pepsinogen is present. What else? Intrinsic factor is present. Mucus is present. Okay, so these are all the important constituents that are present in this gastric secretion. So what are we going to see? Just like our salivary secretion, what are we going to see now in gastric secretion? How are all these components synthesized and secreted into the gastric cavity? What are the functions of these substances that is present in the gastric secretion? How is it regulated? Okay, and then applied aspects related to this. Now that we discussed the salivary secretion, understanding the gastric secretion is going to be way more easier. So we will follow the same pattern that we followed for salivary secretion. First we will look at the functions of the stomach, then we will look at the structures that is responsible for the secretion of all those substances I told you. Okay, then we will understand the difference between a resting and an activated parietal cell. We will look at the composition and function of gastric juice, very important, mechanism of HCl secretion, and then regulation of different phases of acid secretion. So all these points I have listed out can come as an individual short note. Okay, and composition and functions of gastric juice along with the mechanism of HCl secretion with a note on any of the applied aspect can come as a long question. Okay, and multiple MCQs and clinical based questions are also possible from this topic. As we go through the lecture, I will tell you what are the questions that is important for MCQ, what are the questions that will come as a clinical reasoning question, and short notes as well. Okay, even if you look at the PPT, this PPT has been prepared in such a manner that when you download this PPT and when you read, you will be able to answer the questions as you are going to answer in your theory paper. So based on how we evaluate our theory paper, the content of this PPT is also prepared keeping that in mind. Okay, so this PPT can form a base upon which you can write your essay or short note. Okay, so first let us look at the functions of stomach. So there are 7 important functions of stomach, right? So first function, what is the first important functions of stomach? Temporary storage of food. So normal gastric capacity is around 0.5 liters. The normal gastric capacity is around 0.5 liters. When you take a full meal, full stomach, when I say my stomach is full, you are going to hostel, from hostel you are going home for your weekend or to your vacation. What does your mother do? She feeds you to her heart's content, right? So when your mother feeds you to your heart's content, your stomach also becomes full. So how much volume would have reached? Around 1.5 liters. When you say my stomach is full, that means the gastric capacity has reached up to 1.5 liters. Okay. Suppose it is your most favorite food, you keep on eating. What is the maximum to which you can eat? Your stomach can accommodate up to 4 liters of food and fluid, the maximum. So that means what? Normally it is around 0.5 liters. Maximum it can go up to 1.5 liters. Okay, and that is when I say 1.5 liters means that is your stomach is full, and maximum capacity to which you can expand your stomach is 4 liters. Okay, here it is not 0.5 liters, sorry, it is around 50 ml. during fasting state. So during fasting state, the gastric capacity is around 50 ml. When you are fully fed, it can go up to 1.5 ml, 1.5 liters, and maximum you can stretch your stomach is up to 4 liters. Okay. Now, that is why we call the stomach acts as a temporary reservoir of food. It stores the food. Okay. Does it just stop at storing the food? No. Once you have had your food, once it temporarily acts as a storage reservoir of food, then what it is going to do? The gastric secretions are going to come. Okay. Now, once the secretions are going to come, what it will do? It will mix the secretions along with the food. Okay. Through the GI motility, two types of GI motility happens within the two type of gastric motility happens in the stomach. One is called as a retropulsive movements. The other is called as a propulsive movement. Okay. Propulsive movements means what? Pushing the food forward. Retropulsive means what? Pushing it backwards. So by pushing the food backwards, front and back, there are two sphincters, right? The lower esophageal sphincter, the upper. I'll just draw it here. You have the lower esophageal sphincter, then you have your stomach, then we have the pyloric sphincter. Okay. So now the lower esophageal sphincter is also closed, the pyloric sphincter is also closed. Now food is being pushed front and back. This is propulsive movement, this is retropulsive movement. Okay. It keeps on getting pushed front and back. So what is happening? The churning or grinding movement of food will happen. So what does the stomach do? It grinds the food. At the same time, it is also mixing the food along with grinding the food. It is mixing the food with the gastric secretions. Okay. So what are all the gastric secretions that is released from the stomach? First and foremost, most important is hydrochloric acid, then mucus. Then also, once hydrochloric acid is released from the stomach, the pepsinogen that is secreted in the stomach will be activated to pepsin. Okay. Now, all these secretions are mixing with the food. Once the secretions are mixed with the food, we don't call it food anymore. We call it as chyme. We call it as the grinded mixed food is now called as chyme. Now, the stomach, very important function. Okay, we already saw 4, 5. The other important function of stomach is controlled emptying of food. You should know what do we mean by controlled emptying of food. Controlled emptying of food is you can have 700 ml of food, or you can have around 1 liter of food, or you can consume 1.5 liters of food, or even 2 liters of food. It can vary according to your mood, according to your appetite, the quantity of food that you consume. But after grinding and mixing it with the digestive juices, the stomach is not going to empty it proportionately into the duodenum. Okay. Irrespective of how much food you ate and how much food is present in the GI, in your stomach, the pyloric end of stomach is going to open only a little bit so that a specified amount of chyme will enter into the duodenum. Okay, irrespective of the content in the stomach, the quantity of content in the stomach, the pylorus is only going to allow a specified amount of food to a specified amount of chyme to enter into the duodenum. Why like this? Because in the what is the function of the small intestine? Maximum site of absorption. So once the digested food is going to enter into the small intestine, you want it to get absorbed completely. Further digestive process also happens in the small intestine, right? The succus entericus, the pancreatic secretion, everything is going to act upon the chyme. It is going to cause digestion as well as absorption. So for maximum digestion and absorption to happen, the food has to be emptied into the duodenum at regular intervals in a specified amount. So that is called as controlled emptying of food. Then comes other functions of hydrochloric acid. Okay. Now this hydrochloric acid secreted from the stomach, I told it converts the pepsinogen secreted into the stomach to the active pepsin. This peptin is going to cause digestion of proteins. Okay. And this hydrochloric acid provides the proper acidic environment for the full functionality of the pepsin. So pepsin is active in acidic environment. Okay. And this hydrochloric acid which is secreted is also very important for converting the ferric form of iron to the ferrous form. Okay, only when the iron is in the ferrous form it will be absorbed from the small intestine. Okay, so for that absorption to happen it has to be converted. Okay, and also the hydrochloric acid hydrolyzes the iron that is present in the food, so that this iron form complexes with ascorbic acid, vitamin C. Again, vitamin C increases the absorption of iron. Finally, we know the pH of HCl pH is around as less as 0.7. So at this pH no microorganism will be able to survive. So it also kills the microorganism. These are broadly the important functions of our stomach. Okay, so now if you look at the functional anatomy of the stomach is divided into 4 regions. So what you see here in the blue color is your lower esophageal sphincter and cardia. So this region is the lower esophageal sphincter and cardia. Okay, what is the importance of this region? In this region you have secretions that is rich in mucus and bicarbonate. So basically it is going to regulate the acidity of the stomach content. Okay, it is going to neutralize the acidity of the stomach content. And also here what you have is the lower esophageal sphincter, which prevents the reflex of the contents from the stomach into the esophagus. Okay, then the next part, that yellow color part, what you see here is this is the fundus and body of stomach. This is the fundus and body of the stomach. So what is happening here in the fundus and body of stomach? This acts as that reservoir which we are talking. This region basically acts as the reservoir for food, and also all the enzymes, secretions are secreted in this portion. That is, the fundus and body is only having the secretions of all the enzyme, digestive enzymes. Now coming to the last part, that is the antrum and pylorus. So this is the antral region and this is the pylorus. So here again, similar to your cardia, here you have the mucus and bicarbonate secreting cell, and it is the contraction of this pylorus which helps in this grinding. Retropulsive action is also possible, and this pylorus is important for regulating the controlled emptying, and also for one more function. What did I tell you? For the retropulsive movement. Retropulsive movements means for the mixing and grinding movements. All is possible at this region. Okay, so functionally the stomach is divided into the lower esophageal and cardia, then the fundus and body, the antrum and the pylorus. And you know each of this portion of the stomach, what is their function. Okay. Now we will move on to the secretions. That is what is the most important topic of our concern, right? Secretions. What did I say again? Please list it out to me. What are the things that is secreted into the stomach? First and foremost, yes, you're right, it is hydrochloric acid, then pepsinogen, then mucus, then intrinsic factors. Okay, all these are secreted by certain cells that are present. Okay, these cells are present in a specific region called as glands. Since these glands are present in the stomach, we call them as gastric glands. Since these glands are present in the stomach, we call these glands as gastric glands. Now these gastric glands are going to contain those specific cells. Basically these are epithelial cells actually. So these specific cells are going to secrete all those enzymes that I just mentioned, right? Now these gastric glands, depending upon their location, if the gastric gland is present in the cardiac region, we call them as cardiac gastric gland. If it is present in the fundus and body, most important, we call it as oxyntic or parietal or principal gastric glands. If they are present in the pyloric region, we call them as pyloric glands. So what is why are we, all these are gastric glands, then why should we again subdivide them based on the location where they are present? Because if you just look at the functional anatomy, I told you each region has a different type of secretion, and based on the secretion in that region, the function of the region is also varying. That is why How are the secretions different in different parts of the stomach? Because of the presence of different gastric glands. The gastric glands that are present in the cardiac region, that is the cardiac gastric glands, are going to secrete content that is rich in mucus and bicarbonate ions. The gastric glands that are present in the body and fundus are also known as oxyntic glands, are going to secrete contents that is rich in acid, okay, and also pepsinogen intrinsic factor, okay. Then the contents that is coming out from the pyloric gland, which is located in the pyloric region, is again going to produce mucus and also gastrin, mucus and gastrin. So this is important, okay. Cardiac glands is for mucus, oxyntic gland is for mainly acid, pepsinogen and intrinsic factor. The pyloric gastric glands are for mainly mucus as well as gastrin, okay. Now do you understand why, though all are gastric glands, they are located in different locations, so different names? Since they are located in different location, the type of secretion that comes from them predominantly is also going to vary, okay. Now let us look exactly where these gastric glands are present, okay. So what you see here is the empty stomach. This is your empty stomach, okay. It doesn't have food. Can you see all these folded regions in the stomach? So these foldings, what are these foldings? Large foldings called as, these are called as gastric rugae. And now please recall and tell me, the gastric rugae is due to contraction of which muscle? Is it the muscularis externa or is it the muscularis mucosa? Yes, you're right. It is due to the contraction of the muscularis mucosa, the third layer of the mucosa layer. These rugae are present. And tell me, what is the function of these rugae? Gastric rugae, it increases the surface area for digestion and absorption, okay. Now in this gastric rugae, you can see some pits, you can see some invaginations. So the invagination which you see on the surface of the mucosa of the stomach is called as a gastric pit, okay. So this region is the gastric pit, okay. When you go deep into this gastric pit, when you go deep into the gastric pit, you can see these gastric glands, okay. These gastric glands are basically flash shaped structure, okay. So this is the surface epithelium, this is the neck, okay, this is the body, this is the base of these gastric glands, okay. Now where are the cells lined? The cells are lined in this single layer of epithelial cells are forming the gastric gland. Now secretion from the gastric gland, where will it go? It will be emptied into the gastric pit. From the gastric pit, where will it come? It will come to the lumen of the stomach, okay. Now do you understand? So these are the gastric glands, deep invaginations. The cells are present in the gastric glands, different cells, different secretions. All of them will be emptied into the gastric pit. From the gastric pit, it is going to come into the gastric lumen, okay. Now let us look at one single gastric gland and the cells enlarged here, okay. This gastric gland could be a, what are the different types? I told you 3 types of gastric glands. What are those? Please recall and remember and tell me. So if they could be present in the pyloric region, then we are calling as them pyloric gastric gland. If they are present in the cardiac region, we call them as the cardiac gastric gland. And the majority one that is present in the fundus and body are called as oxyntic gastric glands, okay. Now this is a gastric gland, okay. Let us take this to be a oxyntic or a parietal gastric gland, okay. So as I told, this is the neck region, this is the body, this is the base, okay. Now I told you single layer of epithelial cells, but different types of cells are there, okay. At the surface you have something called as surface mucus cell, okay. And then in this neck you have the mucus cell of the neck. Surface mucus cell, neck mucus cell, both these cells are going to secrete mucus. The surface mucus cell as well as the mucus cell present in the neck. Both are mainly going to secrete mucus. Only difference is the surface mucus cell. Mucus is secreted by the surface epithelial cells and also by our neck cells. Okay. The neck cells and the surface epithelial cells are there. The mucus that is secreted by the neck cells are going to be soluble mucus. The one that is secreted by the surface cells are is insoluble mucus. Okay, just for now you remember the type of mucus. The surface cells are secreting the mucus that is insoluble. The neck cells are secreting mucus that is soluble. When I say soluble means what? The mucin gets dissolved in the hydrochloric acid. Insoluble means it is not dissolved. Okay. The insoluble mucus is going to hold bicarbonate with it. So since the insoluble mucus hold bicarbonate with it, it can neutralize the acid. Okay. The soluble mucus cannot neutralize the acid, but it can still form a protective barrier. It can act as a lubricant. Okay. I will tell you again. What we are looking at is a gastric gland. Gastric gland is made up of different types of cells. Each of the cell is going to produce one component of the gastric juice. As we already saw, gastric juice is basically made up of hydrochloric acid, mucus, intrinsic factor and pepsinogen. 4 important components. Okay, please don't forget this. Hydrochloric acid, intrinsic factor, mucus, okay, and pepsinogen. Along with it, other important substances are also there like gastrin, histamine, somatostatin. Okay, but at least briefly you have to remember these important substances. Okay, first we are talking about mucus. So what type of cell is present in the gastric gland that secretes mucus? Mucus is secreted by 2 types of cells. One is the surface epithelial cell, which is present at the surface. The other cells are the neck cells, which also produce mucus. The difference is the surface epithelial cell produces mucus that is insoluble. The neck cell produces mucus that is soluble. What is the function of the soluble mucus? The soluble mucus acts as a lubricant. The insoluble mucus traps bicarbonate into it. Since it is able to trap bicarbonate into it, it causes neutralization of the stomach acid, acidic content. So because of that, the insoluble mucus helps in forming the mucosal epithelial barrier. So the insoluble mucus helps in forming the mucosal epithelial barrier. What is mucosal epithelial barrier? We will discuss during discussing acid peptic disease very shortly. This much is clear. So now we know what are all the cells that is present in the gastric gland that secretes mucus. Then let us come to the from now we finish the surface of the gastric gland, then we finish the neck of the gastric gland. Let's move downward. Now let us come to the body of the gastric gland. The body of the gastric gland mainly contains parietal cells, also called as oxyntic cells. So these parietal cell is going to secrete 2 important substances. One single cell, but it is producing 2 important substances. One is the gastric acid or the hydrochloric acid. The other is the intrinsic factor. What is the function of intrinsic factor? Intrinsic factor helps in absorption of vitamin B12 from the terminal ileum. That means intrinsic factor is secreted by the parietal cells in the stomach. Only if this intrinsic factor is present, vitamin B12 will be absorbed from the terminal ileum. If this is not there, what will happen? Vitamin B12, that is intrinsic factor, is not there. Vitamin B12 will not be absorbed from the terminal ileum and the patient will end up having pernicious anemia or megaloblastic anemia. Then gastric acid. We will see shortly what are all the, just now also we saw what are the functions of HCl, right? So HCl is very important to activate pepsin and kill bacteria. The stimulus for this HCl secretion is 3 important hormones: acetylcholine, gastrin and histamine. Don't confuse, we will see about this in great detail. Okay, so that completes our body of the gastric gland, where we have the parietal cell which secretes hydrochloric acid and intrinsic factor. Then you keep moving down. Come to the base, mainly in the basal region. In the basal region, you have something called as chief cells. In the basal region, we have the chief cell which secretes pepsinogen, the inactive form, which gets activated to pepsin by HCl, and that helps in digestion of protein. Not only pepsinogen, the chief cells also secretes gastric lipase. The chief cell also secretes gastric lipase, which helps in digestion of fat. Okay. Then other cells are also there. Example, enterochromaffin like cells. These are not enterochromaffin cell. Enterochromaffin like cells which is present in the body, which secretes histamine. Okay, what is the use of histamine? Histamine stimulates the gastric acid secretion. That means histamine will stimulate the parietal cell to produce acid. Okay, then we have the G cells. Okay, G cells, again near the base. G cells is very, very important. G cell produces gastrin. What is the importance of gastrin? Gastrin is the most potent stimulator of parietal cells. Just like histamine, gastrin is also going to stimulate the parietal cell. But compared to histamine, gastrin is the most potent stimulator of parietal cell. Okay. Then we also have cells called as D cells which secrete somatostatin. Okay. Remember somatostatin to be a universal inhibitor. Somatostatin, what is the function? It inhibits the gastric acid secretion. Okay. Now I hope it is the gastric glands, the type of cells that is present in these gastric glands and the type of secretions that comes from each of these glands is clear, because this is very, very important. Before proceeding to the next discussion, you should be very clear with this concept. Okay. Now we are going to look into the mechanism of HCl secretion. So already you know what is this? This is a gastric gland, and these gastric glands have different epithelial cells. Each of these cells are different types of cells are there, right? Surface epithelial cell, neck cell. What do they secrete? Mucus. Very good. Insoluble and soluble mucus. Then we have the body of the gastric gland, where you have the oxyntic or the parietal cell. What do these cells secrete? Two important substances, HCl and intrinsic factor. Then we have the G cells at the base which secretes gastrin. Then D cell secretes somatostatin. Then ECL cell secretes histamine. Is this clear? This should be very, very clear. Now let us move on to mechanism of HCl secretion, how HCl is secreted by the parietal cell. Here you are seeing two parietal cells. This is a parietal cell A, B. This is a parietal cell when it is at rest. This is a parietal cell when it is active. What is the difference? When the parietal cell is at rest, you are able to see the tubules and vesicles. You are able to see the tubules and vesicles. And microvilli are very, very rudimentary. When the cell is active, what happens? All the tubules and vesicles are fused. All the tubules and vesicles are fused, and you have a large surface area. You have a large surface area where acid can be secreted. Large surface area where acid can be secreted. And the activity for acid secretion, you need something called as hydrogen potassium ATPase. For acid secretion, you need hydrogen potassium ATPase. This quantity of hydrogen potassium ATPase increases. Okay, so that is the difference between a parietal cell when it is at rest and when it is activated. Okay. Now, okay, the different cells are there and the different substances that is secreted from them. All when it is put together, it forms the gastric juice, right? So again, just like any other gastrointestinal secretion, gastric juice is also made up of majority of water and 0.5% of solids. Solids are organic and inorganic substances. Inorganic substances are mainly the cations, anions, along with the hydrochloric acid, the concentration of which is up to 150 milliequivalents per liter. Under the organic substance, you have the other enzymes, namely the pepsin, renin, gastric lipase, gelatinates and urease. and organic substances like mucus and intrinsic factor. Okay. Now let us look at the functions of these constituents, that is functions of these enzymes that is being secreted in the gastric juice, that is present in the gastric juice. Where does this pepsinogen is secreted from? Pepsinogen is secreted from the chief cells, or the, it's also called as the chief cells or the peptic cells. Actually two types of pepsinogen, this is important for your MCQ. Actually two types of pepsinogen is present. Type 1 pepsinogen, type 2 pepsinogen. Type 2 pepsinogen is produced by all the three types of gastric glands. Type 2 pepsinogen is produced from all the three types of gastric glands, whereas type 1 pepsinogen is produced only from the oxyntic or the parietal gastric gland. That is the difference. Rest of the things you know, I have already told you. Pepsinogen is converted to pepsin, the active form by HCl. Pepsin helps in breakdown of proteins, and the optimal pH where pepsin can break down protein is an acidic pH of 2 to 4. Then the next component is renin. You can see double N here, renin, not renin. This renin is also called as chymosin. This renin is a protein digesting enzyme specially found in animals, not in humans. Mucin, already I told you the insoluble and soluble mucin. We will move on to intrinsic factor. This also we discussed, important for absorption of vitamin B12 from the terminal ileum. Okay, now we are moving to another very important concept, how HCl is secreted. We already saw about the rest of the constituents. Now we are looking at mechanism of HCl secretion, how hydrochloric acid is secreted. Okay, so this is a picture depicting the steps involved in secretion of hydrochloric acid. So where are we going to start? We will start here. This is the first step. Carbon dioxide combines with water to form carbonic acid in the presence of the enzyme carbonic anhydrase. Where do you get carbon dioxide in water? As a result of metabolism. Parietal cell is also a metabolically active cell. As a byproduct of metabolism, you have carbon dioxide and water. They combine to form carbonic acid. This carbonic acid is a very, very weak acid or volatile acid. So carbonic acid, in the presence of the enzyme carbonic anhydrase, carbonic acid is formed. Since it is a weak acid, it will immediately dissociate into bicarbonate and hydrogen ion. For mechanism of HCl secretion, what is needed? For HCl secretion, what do you need? H plus Cl minus. So already now H plus is formed. Now we want, now this, where is the H plus formed? Inside the parietal cell. Now this hydrogen ion, where do you want to go? Do you want to go into the gastric lumen, or do you want it to go into the blood? ISF means interstitial fluid. ISF means interstitial fluid, that is towards the blood. This is the gastric lumen. Now you first decide the direction in which this hydrochloric acid is supposed to go. Yes, you are right. The hydrochloric acid has to go in this direction, that is into the gastric lumen. What is taking it into the gastric lumen? Already I told you, when the cell is active, what is level is increased? Hydrogen, potassium, ATPase activity is increased. When I say hydrogen, potassium, ATPase, what type of transport it is? Primary active transport. Okay. The moment we say hydrogen ATPase, it means it is primary active transport. So hydrogen potassium ATPase is going to actively transport hydrogen ions against its concentration gradient, from the parietal cell into the gastric lumen in exchange for potassium. So hydrogen ion is already pumped out. One of the constituents of HCl is already formed. Okay, now what is needed? Chloride is needed. So in exchange of this hydrogen ion, what has entered into the cell? The potassium has entered into the cell. Okay, the potassium that is entered into the cell, and also on the basolateral side towards the blood, you see the sodium potassium ATPase pump. What is this pump doing? It is pumping 3 sodium ions and 2 potassium ions inside. Already 2 potassium ions is coming inside. Here is another 1 potassium ion. So the potassium concentration inside the cell is increasing. Once the potassium concentration inside the cell increases, automatically it will diffuse out of the cell. Potassium is diffusing out of the cell into the gastric lumen. Now, this potassium is reused, recycled again to pump hydrogen ion out, and this is in. So, this is recycled for the pumping of hydrogen ion outside. Can you understand this much of steps? Basically, with the help of hydrogen potassium ATPase pump, hydrogen ion is pumped out into the gastric lumen. This much is clear. Then you want chloride to enter into the gastric lumen. For that, this is the first step, this is the second step. Okay, then let us look at the third step. This is the third step. The bicarbonate which is formed will enter into the blood. In exchange for this bicarbonate, you have an exchanger here. What is the name of the exchanger? Bicarbonate chloride exchanger. So the bicarbonate chloride exchanger is going to exchange chloride for a bicarbonate. So bicarbonate enters blood. Chloride enters the cell. Chloride enters the parietal cell. After entering into the parietal cell, it is going to diffuse via chloride channel. Through chloride channel, the chloride that entered into the cell is going to diffuse out into the gastric lumen. So this chloride is going to combine with the hydrogen ion to form HCl. This is how HCl is secreted into the gastric lumen. Okay, now I hope you have understood this well. Okay, 3 simple steps. First step is formation of bicarbonate and hydrogen ion from carbon dioxide and water. After that, second step is by the activity of hydrogen potassium ATPase, hydrogen ion is pumped out into the gastric lumen. Third step is the bicarbonate which was formed from the carbonic acid will be exchanged for a chloride on the basolateral membrane. So what is happening? Bicarbonate is entering the interstitial space. From there it will move into the blood, and the chloride ion will enter into the parietal cell. The chloride ion is going to diffuse via the chloride channel into the gastric lumen. The hydrogen and chloride combines to form hydrochloric acid. Okay, so very simple 3-step mechanism where hydrochloric acid is formed. Then we have a very important concept of postprandial alkaline tide. This is again a favorite question of the examiner. They'll ask you, what do you mean by postprandial alkaline tide? See, if you look at this steps, it is very clear. For every hydrogen ion that enters into the gastric lumen, one bicarbonate is entering into the blood, right? So for each hydrogen secreted into the gastric lumen, bicarbonate is reabsorbed back into the plasma. So following a meal, what is going to happen? Acid secretion is going to increase. So following a meal, when the acid secretion increases in the stomach, the pH of the blood is also going to increase. How the pH of the blood is going to increase? When I say pH of the blood is going to increase, that means the blood is becoming alkaline. pH of the blood is increasing means the blood becomes alkaline. Why? Because of the bicarbonate that is reabsorbed. Now, when you filter an alkaline blood, how is the urine going to be? The urine is also going to be alkaline. So following a meal, the urine is alkaline. This is called as postprandial. Postprandial means after a meal, after intake of food, the alkalinity of the urine increases. This is called as postprandial alkaline tide. Okay, very easy, simple concept. Okay, so that is called as postprandial alkaline tide. So now we saw how HCl is secreted. What are all the factors that influence the HCl secretion? So the factors that influence HCl secretion could be hormonal, okay, neural, and also additionally luminal factors as well. So like any gastrointestinal secretion is going to definitely have a nervous factor, a hormonal factor. When we saw salivary secretion, it was purely neural. Okay, there was no hormonal factor involved. But when it comes to gastric acid, we have definitely neural factors affecting it. We have the hormonal factors affecting it. Additionally, certain local factors present within the lumen is also affecting it. First, let us look at the different important hormones that is going to, rather than calling them as hormones, important substances that is secreted from the different gastric cells that is having an action. Okay, so actions of the hormones that is actually present within the stomach. Okay, so among these three, please remember acetylcholine, gastrin, histamine. Okay, all these three, what are these things? Acetylcholine, gastrin and histamine. Okay, so first and foremost is acetylcholine. How is acetylcholine released? Acetylcholine is released from the cholinergic nerve fibers. Acetylcholine is released from the cholinergic nerve fibers. So on stimulation of the cholinergic nerve endings, acetylcholine will be released. This acetylcholine will act on the M3 receptors that is present in the parietal cell. So what is this? This is a parietal cell. Okay, what are we basically seeing? We already saw the parietal cell has hydrogen potassium ATPase, which is the most important factor for hydrogen ion secretion, right? So now this parietal cell, what are all the factors that is influencing this parietal cell to secrete hydrogen ion? It is neural, it is hormonal, liminal. Let us first look at the hormonal factors. So when we look at the hormonal factors, first is acetylcholine. Acetylcholine will be released in response to cholinergic stimulation. That is, vagal stimulation causes the release of acetylcholine from the cholinergic nerve endings. Acetylcholine acts on the M3 receptors, muscarinic receptors present on the surface of the parietal cell. Okay, what it is going to do? By combining with the M3 receptor, it will form hormone receptor complex. So M3 means muscarinic receptor 3. So this M3 receptor upon activation will increase the intracellular calcium concentration. Then comes gastrin. Gastrin, where is it produced from? The G cells. So gastrin is produced by the G cells in the gastric gland. The gastrin that is produced from the G cells of the gastric gland will act on the gastrin receptor. Now the stimulation here is non-cholinergic. It is a vagal stimulation, but it is non-cholinergic. So it acts on the non-cholinergic neuron. It causes the release of gastrin releasing peptide. Via non-cholinergic neurons causes the release of gastrin releasing peptide, which will causes the release of gastrin. This gastrin will act on the gastrin receptor. Once the gastrin acts on the gastrin receptor, it is going to activate the second messenger, inositol phosphate 3. IP3 is going to increase the calcium concentration. Then we have the third important hormone, histamine. How is histamine going to act? Where is histamine secreted from? Histamine is secreted from the enterochromaffin cell. The same cholinergic stimuli which causes the release of acetylcholine via the vagus nerve, the same cholinergic stimuli will also act upon the enterochromaffin-like cells to release histamine. Histamine will act on the H2 receptor. Okay, and it is going to increase the activity of the second messenger, cyclic AMP. Whether it is activation of cyclic AMP or inositol 3 phosphate or directly calcium, what it is going to do? All these second messengers are going to increase the activity of protein kinases. Protein kinases will cause phosphorylation and increase the activity of hydrogen potassium ATPase. Okay, so there are three very important hormones that stimulate the production of HCl from the parietal cells. Okay, acetylcholine, gastrin, histamine. Among these three things, if they ask you which is the most potent stimuli for production of HCl by the parietal cell, which will you choose? It is gastrin. Okay, so among these three, it is the gastrin which is the most potent stimuli for HCl secretion from the parietal cell. How gastrin is stimulated? How the release of gastrin from the G cell is stimulated? That is through the non-cholinergic stimulation by releasing gastrin releasing peptide. Clear? Okay. Now those were the substances that stimulate the gastric. Now what are all the things that inhibit? What are all the hormones that inhibit the production of hydrochloric acid? First and foremost, somatostatin. Somatostatin is a universal inhibitor. It is a universal inhibitor. It is produced by the D cells and present in the gastric gland, and that D cell secretes somatostatin. This somatostatin will act on its receptor and inhibit the formation of cyclic AMP, thereby reducing HCl secretion. Similarly, prostaglandin E2. Where have you heard about prostaglandins? Prostaglandins are formed as a result of cyclooxygenase pathway, right? So through the cyclooxygenase pathway, prostaglandins are all mediators of inflammation. So now this prostaglandin, what it is going to do, it will act on its receptor, and prostaglandin will also inhibit the formation of cyclic AMP. Okay, somatostatin and prostaglandin inhibit the formation of cyclic AMP. When cyclic AMP production is reduced, what will happen? The activation of hydrogen potassium ATPase will be decreased, so acid production will be reduced. So three hormones that increases the acid secretion are gastrin, histamine, acetylcholine. Two hormones that is going to inhibit the secretion of acid are prostaglandins and somatostatin. Okay, this is very, very important. Keep them in mind. Other than this, blood-borne factors like epinephrine can also increase the acid secretion. Finally, what are all the luminal factors? Luminal factors means factor that is present within the gastric lumen. Example, distension of the stomach, the physical stretching of the stomach due to the entry of food. And the products of protein digestion, when pepsin acts on the proteins present in the food, the peptides and amino acids that are released, they act as stimuli for acid secretion from the stomach. Now, what are all the factors that inhibit? Already we saw the hormonal factors. Luminal factors, when the chemical composition, the acid level increases, when the chyme becomes acidic. This is example of negative feedback mechanism. When the acid level increases, what it is going to do? It is going to inhibit its own secretion. Then blood-borne factors like secretin, gastric inhibitory peptide and glucagon, all these are factors that is released from the small intestine. So small intestinal hormones is going to inhibit the production of HCl from the stomach. Okay, easy to remember. Now coming to the different phases of gastric acid secretion. Generally what are the different phases of gastric acid secretion? We have the cephalic phase, we have the gastric phase, then we have the intestinal phase. Be it for gastric acid or be it for small intestinal and large intestinal secretion. Any of these secretion, we have the gastric, we have the cephalic, we have the gastric, and then we have the intestinal phases. First phase is the cephalic phase of gastric acid secretion. Why do we call it as cephalic phase of gastric acid secretion? Because the stimuli to produce gastric acid is coming from the head or the brain. That is why it is called as the cephalic phase of gastric acid secretion. How much does this concentration? 40 to 50% of HCl secretion is because of this cephalic phase of gastric acid secretion. Okay. The idea of giving an appetizer. Can you name some appetizers? Yes, all those starters that you eat. Soup and starters before when you eat a 3-course meal, 5-course meal, 7-course meals, what is the first thing? First they will give you soups and starters. These are basically appetizers. I will come back to this. Basically, what are these appetizers doing? The appetizers are basically going to increase the acid secretion from the stomach. Okay, how? Once you slowly drink the soup and eat these appetizers, it is going to increase the acid secretion. When the acid secretion increases, basically what is happening? The stomach is preparing itself for the meal that is going to follow. Okay, so that is the idea of giving appetizer. And why is it called as appetizer? Because it not only prepares the stomach for the meal by producing HCl, it also increases the appetite of the person. Okay, the secretion of HCl will increase the appetite of the person. So that is why they are also called as appetizers, the soups and starters which is given at the starting of the meal. So how are we supposed to have the soups and starters? You have to take it slowly. Okay, you have to enjoy it. When you take it slowly, what is going to happen? It will stimulate your appetite, keep you ready for the things that is going to follow, and that is psychologically. Then what is it going to do? Even physiologically, in the gut, in your stomach, it is going to increase the acid secretion so that it will be easier for the stomach to digest the meal that it is going to receive. Okay, now coming to the cephalic phase. So cephalic phase contributes 40 to 50% of the HCl that is secreted in the stomach. In cephalic phase, what is going to happen? The same, through conditioned and unconditioned reflex. Okay, what is going to happen? The limbic system, hypothalamus and the vagal nuclei which is located in the medulla, all this is going to initiate a vagovagal reflex. Vagovagal reflex already I discussed when we were discussing the functional anatomy of the gastrointestinal tract. So it is going to stimulate a long reflex, okay, which will act on the enteric nervous system. By acting on the enteric nervous system, what it will do? It will stimulate the enterochromaffin like cell to secrete histamine, parietal cell to secrete acid. As I already told you, once the acid level increases, it has its own negative feedback and it inhibits the parietal cell. And also, increase in acid as a negative feedback will also inhibit the G cell so that gastrin is not produced. That is when the HCl level increases beyond the physiological limit. That was the cephalic phase of gastric acid secretion. Then let us move on to the gastric phase of gastric acid secretion. That is what happens in the stomach. So for the gastric phase of gastric acid secretion contributes 50 to 60% of total HCl that is produced. Okay, 40 to 50% is contributed by the cephalic phase, easy to remember. Next, 50 to 60% of the HCl synthesis is contributed by the gastric phase of gastric acid secretion. In this gastric phase, cephalic phase is purely neuronal, whereas gastric phase is both neuronal as well as certain local factors. Let us look at the local factors. First let us look at the local factors. Local factors is distension of the stomach and products of protein digestion in the stomach. When these things are present, what will happen? It will create a local reflex. And through this local reflex, what is happening? The same thing what I told you. The G cells, all these products of protein digestion and distension of the stomach will activate the mechanosensitive and chemosensitive receptors. Okay. Then the G cells will secrete gastrin. Gastrin will cause release of acid from the parietal cell. Okay. Only difference what you have to note down between the cephalic phase and gastric phases. In the cephalic phase, it is a long reflex. In the gastric phase, it is a local enteric reflex. Okay. Now coming to the last phase, that is the intestinal phase of gastric acid secretion. This intestinal phase actually contributes only to the 10% of HCl that is secreted in the stomach. Even when we are talking about this intestinal phase, initially it increases the secretion, but overall intestinal phase decreases the HCl secretion. Please remember, this is an important concept which I don't want you to forget. Okay. When it comes to intestinal phase of gastric acid secretion, initially the 10% of HCl that is secreted is because of the initial phase. But overall, when you consider the intestinal phase, at the end, during intestinal phase, actually the hydrochloric acid secretion is inhibited. So how does it happen? First, how does the initial stimulation happen and how does the actual inhibition happen? Initial stimulation, the same thing like that of our gastric phase, that is presence of substances of protein digestion in the duodenum, and distension of the duodenum is going to cause a vagovagal reflex, increasing the HCl secretion in the stomach. Okay. And at the same time, it is also going to cause the release of gastrin from G cells in duodenum as well. This is the initial phase. Okay. Later on, what will happen? A local enterogastric reflex will get activated. How enterogastric reflex is getting activated? The same thing, distension of the stomach and presence of acidic chyme in the duodenum. Acidic chyme in the duodenum will activate enterogastric reflex. What do you mean by enterogastric reflex? This is gastric, this is entero, that is the duodenum. So, presence of acidic chyme and products of protein and fat digestion, all these things will go and inhibit the parietal cells to secrete acid. Why it is inhibiting the parietal cell? Why is it important? Other two phases, acid secretion is actually increasing in cephalic phase and gastric phase. Whereas in intestinal phase, except for the initial part, overall what it is doing, it is only inhibiting the gastric acid secretion. Why? Because it inhibits the gastric acid secretion, it also inhibits the gastric motility. Okay, inhibits the HCl secretion from the parietal cell. Why? Because otherwise what will happen? That controlled emptying of stomach, I told you, that one function, controlled emptying of stomach, that will not be possible. All the gastric contents will be emptied into the duodenum at one go, which is going to hamper the functioning of duodenum. That is why the acidic chyme is going to activate the enterogastric reflex, where duodenal stimulation will inhibit the acid secretion from the stomach and also inhibit the gastric motility. So that slowly, little chyme will be entering into the duodenum, so that the process of digestion absorption in the intestine happens smoothly. So what are all the GI hormones that is released in the duodenum that inhibits this acid and acid secretion from the stomach and gastric motility? It is secretin, gastric inhibitory peptide, vasoactive intestinal peptide, bulbogastrin and enteroglucagon. Okay. So far what did we see? We saw how HCl is, functions of HCl, how HCl is steps involved in HCl secretion, and what are all the nervous, hormonal and luminal factors that regulate HCl secretion. And we saw the different phases of gastric acid secretion, that is the cephalic phase, the gastric phase and the intestinal phase. Now, how I was telling during the gastric phase, this much amount of HCl, this much percentage of HCl secreted. During cephalic phase, this much of percentage of HCl secreted. The nervous regulation has this much of role. The luminal factors has a role. Hormones have a role. All these things I was telling. How did we find out? Through these different experiments, we found out the different phases of gastric acid secretion. So to identify the cephalic phase of gastric acid secretion, or to prove that gastric acid is produced even during the cephalic phase, they did an experiment. A name of that experiment is called as sham feeding. They did an experiment. A name of the experiment is called as sham feeding. What do we mean by sham feeding? Here we have took the experimental animal. Here is a dog, and a fistula is made at the esophagus. A fistula is made at the esophagus. Now what is happening? The presence of food in the mouth will cause, this is a cephalic phase, right? Presence of food in the mouth is going to increase the acid production in the stomach. So to measure this, what they did was, they placed a cannula in the stomach and measured the acid. Placed a cannula in the stomach and measured the acid. At the same time, the food what the dog ate came out of the esophageal fistula. That means the food did not enter into the stomach. Okay, now what they did was only measure the acid that was synthesized in the stomach. So as a result of eating, so just by the cephalic phase, they measured the quantity of the acid and also the other constituents and its properties. Okay, so that is the cephalic phase of experimental evidence for gastric acid secretion. So what is the name of the experimental evidence for cephalic phase? It is called as sham feeding. Now, similarly, we have experimental designs even for study of the gastric phase. Okay, for gastric phase, it is basically pouches. So remember the word pouch. When you remember the word pouch, it refers to the gastric phase of experimental design. 5 different pouches were formed. Different types of pouches were formed. Okay, Pavlov's pouch, Hayden's pouch, Bickel's pouch, Ferrell's pouch, and Ivey's pouch. We are more concerned with Pavlov's pouch. Okay, this Pavlov's pouch is similar to a stomach. Okay, a small pouch was separated from the main body of the stomach. This Pavlov's pouch had all the layers of the stomach. The nerve supply and the blood supply was also intact. Now, whatever was happening in the stomach in response to the food was also happening in the Pavlov's pouch. That is how they studied the neural and chemical factors in gastric, role of neural and chemical factors in gastric acid secretion. Okay. Hayden's pouch. This is also for the gastric acid secretion in the gastric phase. Only difference here is compared to our Pavlov's pouch, in the Hayden's pouch, the neural factors, that is to study the neural influence of neural factors on acid secretion, they denervated the pouch. All the layers are there, blood supply is intact, but the nerve supply was denervated. So since they denervated the nerve supply, now we were able to study the role of neural factors in acid production during the gastric phase. So these are the experimental studies. These experimental studies are also very, very important because that can be asked as a separate short note. Okay. So this is a tabular column which is overall summarizing the major stimuli, their control mechanism and their contribution to the cephalic phase. Once you go through the topic, I would highly recommend you to go through this tabular column so that you will be able to remember and revise whatever you have said. Okay. Because of the time constraint, I am not going back again into this tabular column. You can go through this tabular column again when you have time. Okay. Now coming to the applied aspects. So before closing for the day, let us look at the applied aspects related to increase and decrease the production of hydrochloric acid. What is gastritis? Gastritis is any clinical condition where you have abdominal discomfort due to indigestion or dyspepsia, due to altered level of acid secretion or due to indigestion. 2 types of gastritis there, acute gastritis and chronic gastritis. What are all the reasons for this gastritis? Unhealthy diet, eating junk food, food that is rich in fried, spicy food, personal habits, that is what eating of junk food, severe stress, type A personality, person who is always angry, hungry, hyper, infections, okay, drugs. What do we mean by drugs? Mainly your aspirin and painkillers, chemical and physical agents, and chronic gastritis if left untreated. All this and stress, all this can lead to gastritis. And this gastritis, chronic gastritis, if it is not untreated, then it will lead to formation of peptic ulcer. The first step is gastritis. If gastritis is not treated, the patient is going to end up having peptic ulcer. Okay. Classification of gastritis, acute gastritis and chronic gastritis, already told you. Acute gastritis is usually due to H. pylori infection. Chronic gastritis could be because of an autoimmune pathology or H. pylori or a reflex gastritis. Okay, that is because of presence of chemicals. Then comes our most important topic, peptic ulcer. What do you mean by peptic ulcer? Peptic ulcer means an ulcer either in the stomach or duodenum. An ulcer either in the stomach or duodenum is called as peptic ulcer. If the ulcer is present in the stomach, then we call it as gastric ulcer. If the ulcer is present in the duodenum, we call it as duodenal ulcer. What is the reason? What is the pathophysiology of this peptic ulcer? Its pathophysiology and physiological basis of the treatment is a very, very favorite exam question. Okay, I want you to listen carefully here. So talking about peptic ulcer means it involves both gastric ulcer as well as duodenal ulcer. Peptic ulcer is, what is the cause of peptic ulcer? Either due to the decreased mucosal defense mechanism. We told no mucosal defense barrier. because of the insoluble mucin that is produced by the surface mucous cell. Either that barrier is decreased or increased hypersecretion of acid or an infection, especially H. pylori infection. Any of these three things can be a causative factor for peptic ulcer. Diminished effectiveness of mucosal. This mucosal barrier itself can come as a short note. Okay. So if they ask you about the mucosal barrier, what will you write? First, what do you mean by mucosal barrier? It is nothing but a defense barrier of the stomach that helps in preventing the mucosal epithelium from getting eroded by the hydrochloric acid. So that is the definition. So this is your definition of mucosal barrier. Okay. Now, what are all the key components and their function? So the mucosal barrier is made up of a mucus layer. What is this mucus layer made up of? Obviously, mucin. Okay, it is made up of a thick jelly-like substance called mucin that is secreted from the mucosal cells. This mucin, height of this mucin is around 0.2 millimeter. So on top of the epithelial cell, around 0.2 millimeter of mucin is present. This jelly-like substance is insoluble. Okay, it contains glycoproteins like mucin and bicarbonate. So what do? This mucin and bicarbonate will trap and neutralize the acid that is present on the epithelial surface, and also it is insoluble, so it prevents the gastric mucosal epithelial cells from coming in contact with the acidic luminal content. Okay, so it has three components: the mucus layer, then the mucus cell. Mucus cells are nothing but the specialized epithelial cell which releases this mucus, and also the mucus droplets. Mucus droplets basically contains glycoproteins and bicarbonate which helps in neutralizing the acid. Okay. Then bicarbonate, already I told you, it is because of this bicarbonate is also secreted by the epithelial cells. Okay, so the bicarbonate neutralizes the acid and maintains the pH of the mucosal surface at around 7. Then we have the capillary and interstitial fluid as well. Now what do these capillary and interstitial fluid do? They deliver nutrients and bicarbonate to the mucosal cell, and also it facilitates the exchange of ions. Okay. So here is a picture of your mucosal defense barrier with all the components. Okay. So what do you have? The mucus gel. Okay. Then you have the second component, that is the mucus droplets. Then the third component is the mucosal cell. Fourth is the interstitial fluid, and fifth is the capillary. So all this together forms the mucosal epithelial barrier. Okay. Basically the mucosal epithelial barrier is a jelly-like substance of 0.2 millimeter diameter. 0.2 millimeter in diameter, which is made up of mucin droplets that contain bicarbonate and glycoproteins, and also other nutrients and ions from the blood. Together what do? They neutralize the acid and maintains the mucosal pH at around 7, thus providing the physical barrier to the mucosal epithelial cells so that they don't get damaged by the acidic chyme that is present here. Okay. So that is what physiological significances: protection from the acid by preventing the corrosive effects of the gastric acid. Normally the luminal contents, the pH of the luminal contents is around 1 to 2, but because of this mucosal epithelial barrier and the bicarbonate ion which is present in it, the pH of this epithelial barrier is around 7, and also the disruption of this will cause the occurrence of ulcers. Okay. Now, when can this mucosal epithelial barrier be disrupted? Whenever we use drugs like aspirin and non-steroidal anti-inflammatory drug, what will happen? This non-steroidal anti-inflammatory drugs and all will act via, it will inhibit the cyclooxygenase pathway. It inhibits COX 1 and 2. Cyclooxygenase 1 and 2 will be inhibited. So inhibition of prostaglandin E2 synthesis. So when prostaglandin E2 is not there, increase in HCl production will be there. That leads to the damage of this mucosal barrier, and also chronic stress will lead to stress induced ulcer. Now that was one. So what are all the reasons what are all the reasons an ulcer can form? Ulcer can form because if the mucosal barrier is not properly formed or if it is not intact, then it could be either due to hypersecretion of acid. So now we are looking at the second reason, that is the hypersecretion of acid. What you see is an endoscopic finding of a peptic ulcer. Okay, so when can acid secretion increase? Easy, hurry, worry, curry. Okay, when can acid secretion increase? Whenever we are in a hurry, type A personality, always stressed, worrying, and also when we eat food that is fried, spicy, rich food, all these things can increase the acid secretion. Or certain conditions like gastric outlet obstruction syndrome, Zollinger-Ellison syndrome, also called as gastrinoma. Here also the gastrin secretion increases, which increases the acid secretion. Okay. Or finally an infection like an Helicobacter pylori infection. H. pylori is a gram-negative bacillus which secretes an enzyme called as urease. What does it secrete? Urease. What will this urease do? It will convert urea into carbon dioxide and ammonia. Now this ammonia will neutralize the HCl. Ammonia will neutralize the HCl. Once this HCl is neutralized, what will happen? This Helicobacter pylori will thrive in this region. Usually acid is very potent, it is going to kill all microorganisms, but this Helicobacter pylori produces ammonia which is going to neutralize the acid. Since the level of acid reduces, immediately what it will do? This bacteria will grow. Once this bacteria grows, what it is going to cause? It is going to cause local inflammation. It disrupts the immune response, and additionally it will inhibit the somatostatin secretion from the D cell. If somatostatin secretion is also decreased, what will happen? HCl secretion will be increased. Okay, so that is how Helicobacter pylori infection causes peptic ulcer. Now what is the physiological basis behind the treatment? First line of treatment is usually prescription of a proton pump inhibitor, that is the hydrogen potassium ATPase pump, which is primarily responsible for hydrogen ion secretion into the gastric lumen. So if you block that pump, for example with drugs like omeprazole, acid secretion will be reduced. Next is H2 blockers, antihistaminics like cimetidine, famotidine and nizatidine. All these are H2 blockers which helps in decreasing the release of histamine, so that HCl secretion is, parietal cell stimulation is decreased, so HCl secretion is decreased. Or surface protective wound ulcer healing substances like sucralfate, they also form a protective covering on the ulcer so that it doesn't get eroded further. And then gastrin blocker. Gastrin blockers are still in experimental phase. We don't have a gastrin blocker in market as such, but a very promising gastrin blocker is proglumide. Okay, proglumide is a gastrin blocker because among all these secretions, it is the gastrin which is the most potent stimulator for HCl secretion. So if we are able to block the gastrin, then HCl secretion will be reduced. So an example of gastrin blocker is a proglumide. Then antibiotics like amoxicillin to kill H. pylori. Then also the acid production will be reduced. Whereas muscarinic blockers, remember muscarinic blockers, M3. Actually what did I say? Acetylcholine acts on the M3 receptors and brings about acid secretion. So like a muscarinic blocker like atropine should have been an effective treatment for treating hypersecretion of acid, but this doesn't happen. Okay, atropine is not an effective treatment for hypersecretion of acid. Why? Because as we saw initially, the stimuli for acid secretion, if you remember the slide, I told you the stimuli for acid secretion, HCl secretion stimuli, 3 types of stimuli are there. One is gastrin, the other is acetylcholine, then the other is histamine. In this, acetylcholine and histamine are cholinergic. Okay. Are cholinergic. Whereas gastrin is non-cholinergic. Non-cholinergic stimuli were the release of gastrin releasing peptide. That is why Even when you give atropine and block the cholinergic stimuli, still acid production will be there through gastrin. That is why atropine does not give encouraging results. Okay, this is again a very important viva question. Why doesn't atropine tend to be a promising treatment modality for reducing HCl secretion? Why? What is the answer? Because acid secretion is not entirely dependent on the cholinergic stimulation. Majority of the acid secretion is also dominated by the gastrin release, which is through non-cholinergic pathway, that is release of through gastrin releasing peptide. That is why atropine is not a preferred treatment modality when it comes to treatment of peptic ulcer. Is it clear, this concept? Okay, those are specific treatment. Non-specific general measures, we give antacid, which will cause immediately neutralizing effect of the acid and reducing the pain. And other methods like yoga, good sleep, avoidance of painkillers, avoidance of spicy food and alcohol. When none of these modalities work, sometimes surgical treatment like vagotomy and gastrectomy can also give some relief from increased HCl production. Okay. Gastrectomy, which part of the stomach will they remove? Mainly the antral region. Okay. Now that completes our discussion on the applied aspects. Finally, before discussing our MCQs, let us have a look on the gastric function test. Gastric function test can also come as a separate 5-mark question. So we will look at these tests. What is the purpose of this test? What is the physiological basis and their interpretation? Okay. In a very simple manner, I have tabulated these tests for you. So first test is a basal acid output test. What does it measure? It measures the acid secretion while in the fasting state. What is it used for? To assess the baseline acid production by the parietal cells. It is increased in Zollinger-Ellison syndrome and decreased in gastric atrophy. Okay. Now coming to the maximal acid output. What do maximal acid output actually measure? It measures the acid secretion after stimulation. That is not in the fasting state, in the fed state. What is this going to evaluate? It is going to evaluate the capacity of the parietal cell. Okay. Again, similar to our basal acid output, it will be increased in Zollinger-Ellison syndrome and it is decreased in pernicious anemia. Okay, why it is decreased in pernicious anemia? Because again here, because of the gastric atrophy, there are no parietal cells to get stimulated. Okay, because of that. Then we have the pentagastrin test. What is this test going to do? It is going to stimulate and measure the gastric acid output. How? It is going to mimic, pentagastrin is similar to gastrin. So it is going to mimic the action of gastrin and we are basically testing the function of the parietal cells. An abnormal response suggests gastric hyposecretion or hypersecretion. Then comes the histamine test. What are we going to measure here? Again, you are going to measure the maximal gastric output by using the histamine, which stimulates the H2 receptors. Now, histamine test, the output is going to be increased in duodenal ulcers and it will be reduced in gastric atrophy and gastric cancer. Then we have another test called the tubeless gastric analysis. Here we are estimating the acidity of the gastric contents by using a dye. Okay. When the recovery of the dye is low, it indicates hypochlorhydria or achlorhydria. Okay. Basically, we are going to measure the amount of dye excreted in the urine after the food is getting digested. Then we have the gastrin assay, where we are going to measure the levels of gastrin, especially helpful in Zollinger-Ellison syndrome and in atrophic gastritis. Okay, to differentiate. Then we have pepsinogen assay, which is going to reflect the function of the chief cells. This is again going to be low in case of mucosal damage and high in case of gastric inflammation. Then we have urea breath test, specially employed to detect H. pylori infection. This is positive in case of H. pylori infection. Then Schilling's test is to evaluate vitamin B12 absorption. Okay, low levels is going to indicate that the patient is in pernicious anemia and ileal dysfunction. The vitamin B12 levels is going to be low. Okay. So that is the end of our discussion on salivary and gastric secretion. Again, this is a complete summary of our gastric acid secretion. Once you are done with the entire lecture, again I would suggest please take some 5 minutes, go through this. If you are able to understand all the things what we have mentioned here, that means you understood the topic. And while revising, you can keep this page and revise so that you will be able to recall everything easily. Okay, recall all those things you read. Now quickly let us look at some multiple choice questions and clinical reasoning questions. So here you have first question. A patient complains of dry mouth, xerostomia, and has difficulty in swallowing. Which of the following glands is the largest contributor of resting salivary secretion? Please look at this word, resting salivary secretion. I already told you there also. What is the answer? When we are discussing the composition and functions of saliva, we told when it comes to resting salivary secretion, the answer is submandibular gland. When it is active, it is the parotid gland. When it is resting, it is the submandibular gland. Okay. Second question, which neurotransmitter is primarily responsible for stimulating salivary secretion during parasympathetic activation? So, during parasympathetic, what is it? Cholinergic. Yes, it is acetylcholine. Okay. So, when it is cholinergic, how is it? It is going to be a watery saliva rich in electrolytes and enzymes. A 45-year-old patient presents with persistent dry mouth, increased dental caries, history of an autoimmune disease. Okay. Which condition is most consistent with our findings? What is it? Sjogren's syndrome. So in Sjogren's syndrome we have xerophthalmia, xerostomia. It is an autoimmune condition, right? Which component of gastric juice is responsible for activating pepsinogen? Which is it important for activation of pepsinogen? It's a direct answer. Yes, HCl. HCl converts pepsinogen to pepsin, and pepsin helps in breakdown of proteins. A patient with severe gastroesophageal reflux. A patient with severe gastroesophageal reflux is found to have increased gastric acid production. Identify the hormone and the cell that is responsible for secretion of the hormone. The question might look lengthy, but it's a very easy question. What they are asking? Tell the cell that secretes this hormone that is causes increased gastric acid production. Which among these things is the most potent stimulator for HCl secretion? Yes, it is option B. G cells that secretes gastrin. Next, what is the physiological basis for postprandial alkaline tide? Is it secretion of bicarbonate per parietal cell, or increased secretion of gastric enzyme, or it is the release of gastrin into the bloodstream, or is it the decreased chloride reabsorption? What is the answer? Yes, it is the secretion of bicarbonate by parietal cell. For every acid that is secreted in lumen, one bicarbonate is secreted into the blood, which increases the alkalinity of the blood, thereby increasing the alkalinity of the urine, and this is called as postprandial alkaline tide. Next, which experimental method isolates a gastric pouch while preserving its nerve supply, allowing to study both neural and chemical factors? The moment you see pouch, which phase of gastric acid secretion should come? The gastric phase. Now in this pouch, both neural and chemical are intact. That is, the nerve supply and the blood supply is intact. That means which is it? Pavlov's pouch. Correct. In Hentin's pouch, what did we do? Nerve supply is cut. That is the difference. Okay. Then we have a patient with reduced stomach acid production following surgery that accidentally severed the vagal nerves. Which phase of gastric acid secretion is most affected? When it comes to nervous stimulation, which is the phase? It is the cephalic phase. Yes, direct answer. Then next question, which of the following is true regarding HCl secretion by parietal cell? How do parietal cells secrete HCl? Is it directly stimulated by secretin, or does it involve hydrogen sodium pump? Does it require hydrogen potassium ATPase pump, or it is inhibited by gastrin? Which is the answer? Direct answer again. It requires the activity of hydrogen potassium ATPase pump. Coming to the last question. A 50-year-old male presents with burning epigastric pain. So he has epigastric pain, which is very, very suggestive of peptic ulcer, right? That worsens on an empty stomach and improves on eating. This is also another important finding. Worsens on an empty stomach and improves after eating. Okay. He reports a long history of use of NSAID. An upper GI endoscopy revealed a gastric ulcer. What is the most likely physiological mechanism contributing to this condition? So among the 4 options, which do you think is the answer? Is it because of, so this patient has acid peptic disease. Okay. Already they have confirmed it on the endoscopy as well. He has a gastric ulcer. Just because it is a gastric ulcer only, it is improving after eating. It worsens on an empty stomach and it improves after eating. Now, is it because of increased gastrin production, or it is because of reduced bicarbonate secretion, or is it in this patient inhibition of prostaglandin synthesis, or is it hyperactivity of the vagus nerve? What is the answer? See, the key they have given here is he has been taking NSAID for joint pain. So what is the answer? It is option C. It is the inhibition of prostaglandin which has led to increased HCl, which is reducing the effectiveness of the mucosal barrier. Okay. While discussing the mechanism of HCl factors that is increasing and decreasing HCl secretion, we saw, right? What do prostaglandins do? Prostaglandins inhibit the HCl secretion, correct? Now this NSAID is going to inhibit the cyclooxygenase enzyme, so prostaglandin levels are decreased. When the levels of prostaglandin is decreased, now what is going to happen? The levels of acid is going to increase. More acid, the mucosa lining is going to be more susceptible to acid damage. Okay. Before ending the topic, I want you to remember one more point here. The patient is complaining of relief in pain after eating. Okay. So whenever the patient is having relief in pain after eating, means when the pain improves with food, then it is due to bicarbonate neutralization. Okay. Pain improves means it is because the type of ulcer is a duodenal ulcer. If the pain worsens with food, that means it is called as gastric ulcers. When the patient is eating, that is why here actually the patient reveals a duodenal ulcer. It should be a duodenal ulcer. Okay, not gastric ulcer. Now, why? Because what happens in gastric ulcer, when you eat food, the pain will worsen when you eat food. Why? Because the food is going to stimulate the acid production, and also it is going to cause mechanical irritation of the eroded mucosa. So the pain increases. But in case of duodenal ulcer, what is going to happen? Whenever food enters into the duodenum, what is going to happen? Pancreatic secretion is going to come. The pancreatic secretion is going to be rich in bicarbonate, which is going to cause neutralization. So after eating food, they might have some relief. That is why the actual diagnosis for that given patient is duodenal ulcer and not gastric ulcer. Okay. I hope you had a very thorough, clear understanding of all the concepts that you need to know with regards to the salivary and gastric secretion. When you go through this lecture, I am very confident that you will be able to approach your clinical based questions, your MCQ, your long essay, short essay, your Viva. All these things you will be able to approach with confidence. All the very best. And if you have any doubts, you can still contact us as Dopamine to clear your doubts. Thank you.